Hello, everyone, and welcome back to Creation Myths. All right, as always, let me just make sure everything looks okay on YouTube before we go any further here. I just want to make sure I can hear me and see me. Chat, if you have any audio or visual problems, let me know. I'm reading the chat, I will see it, and I can try to address any issues that might pop up. I'm just waiting for YouTube to kind of cycle here and catch up, and then we'll get going, folks. Come on, YouTube. Just waiting. Need to make sure I can hear me and see me. Okay, I can hear me and I can see me. It looks fine. I think we're good to go. Okay. Let me make sure I can see the YouTube chat. Yes, I can. Yeah, I know. Other than my wrong background. Sorry. I know. Sorry, everybody. It's never coming back. Okay, couple quick housekeeping notes here. So like I said, any issues at any point, let me know. Especially, I am going to be shuffling between a couple different screens we're going to be sharing for this first part. So it might take a couple minutes, like just if there's anything weird with something's not showing up, I'll always tell you what you should be seeing. And if you don't see it, let me know and I'll try to fix it. Also, I'm bad at this. If there are any super chats, I will read them at the end. I know I'm bad at that. I will make sure that I do that. So just FYI, I see you. I will do that. So let me tell you what we're doing here, folks. My guest tonight is Jason Vance. Jason is a commenter on YouTube, and we were chatting in the comments, and I said, look, why don't we just talk about this? Email me. And I will tell you, 99 times out of 100, I say that, and I never get an email. But Jason actually emailed me to set up this conversation. So here we are. So Jason, take a minute, introduce yourself, tell everyone about you, whatever your background, whatever you want to tell people, and then we'll kind of move on.
Okay, sure. Thank you, Daniel. I appreciate it. Well, Jason Vance, I've had an interest in the complexity of life since I first started considering seriously the belief that life has evolved from a simpler life to more complex, and the idea that we're all descended from a common ancestor. I actually believed that for a long time. My dad has 2 degrees, one's biology, one's psychology, and as a little boy, I grew up with a theory, and I never questioned it. And then I got into college and just started questioning whether or not it made sense that mutations, which is the mechanism that evolution says drives all changes, or drives speciation, creates new species, creates new organs, that it made sense that mutations, which none of us want to see mutations in our children, they aren't something that is, they're not positive. That's the reason we have mutation repair mechanisms to correct them. And so the theory just didn't make much sense to me. Then I read a book by Michael Denton, Evolution of Theory in Crisis, and that kind of opened my eyes. And then there's also the fine tuning of the universe, where everything seems evidently, it's clear to me, is designed. And so there's a design, and the design is clear in nature. Its design is clear in the universe and the solar system. So, but that being absolutely clear to me, it's certainly not clear to everybody else, especially biologists. So I've had this just perplexed, this perplexed, not viewpoint, but the perplexity of knowing that what's so obvious to me is not obvious to everybody else has always just kind of befuddled me. But the more I look into it and investigate, the more it seems to me that life is a special creation and did not evolve. And I think one of the things that I took exception to in the comments or the chats that Daniel and I had was the idea that you can go on the internet and find out that natural history says that there was a shrew-like animal that lived 226 million years ago, and all mammals today are descended from that little fella. And so, that means hippos, giraffes, gorillas, humans, whales are descended from a shrew-like creature. And I think genomes first prefer stasis, try to maintain stasis, and stasis means not changing. So that's where I'm at, Daniel, and I'll be happy to share a few things, just a few slides to get us going when you're ready. And I appreciate you having me on too, Daniel. And I'll tell you, one of the reasons I responded and said, "Yeah, put me on," is, man, I'm 70 years old. If I don't need to be shy about what I believe, miss an opportunity to talk to somebody like you, I think, who has a different opinion. That's just important to do. So I'm glad I said yes, because I enjoy getting to know people, and I'm going to enjoy getting to know you too.
Awesome. Thank you.
You're welcome.
So, people in chat are joking of you calling me Daniel because everyone just calls me Dan. You just call me Dan.
Okay.
It's like, yes, that's technically my name, but yeah, you just call me Dan. It's fine. Okay, so I want to make sure, one thing I always do when I have people on is I want to make sure that I understand your position correctly. So, how do you characterize your own position here? Young Earth, old Earth, intelligent design? What's your, like, how do you characterize yourself?
Intelligent design. And I know that there are those, I think, scientists who are proponents of intelligent design. Typically, I think those are old Earth creationists, and I'm not here to dispute the age of the Earth. I think there's good science from what I've seen. Hugh Ross believes it's an ancient Earth, and I think probably other prominent men who believe in intelligent design or a similar mindset. I think Stephen Meyer would say, Richard Sternberg probably line up with an old Earth, and I don't have a problem with that.
All right, so we're off to a great start. You're already, no pun intended, light years ahead of most people, most creationists I talk to who are young Earthers, and then you have to, there's everything is wrong with young Earth. But so if we're starting from the Earth is 4.5 billion years old or so, we're doing great. So I want to show, first thing I'm going to do, everybody, is show the some of the YouTube comments. This was from a video from like last month, and I just want to kind of just to contextualize what we're talking about here. So let me pull these up, and this will kind of give us an idea of kind of what led to this conversation. So this is kind of the key one right here, I think. All genomes from bacteria to humans. Let me just get this window so I can move it. All genomes from bacteria to humans have mechanisms to repair mutations, to keep them from being inherited. It's prima facie evidence that nature seeks stasis in genomes, not change. And then we got this one right here. I was talking about this virus called FIX174, which is selected for a higher mutation rate than it would otherwise have. It suppresses DNA repair. And you responded, as I'm sure you know, there's absolutely zero evidence that FIX174 virus and the E. coli bacteria it infects will ever be anything other than FIX174 and E. coli. Every mutation in FIX and the only bacteria it infects only and always ensure that FIX will be FIX, E. coli will be E. coli forever. Knowing this is true, why would you believe that hippos, elephants, giraffes, etc. evolved from ancient shrews? Okay, so there's kind of two levels to this. There's the DNA repair mechanisms seem to cause a problem for evolution because they work against change, right? They cause stasis, and you need change for evolution. And then on the more kind of macro scale, right, we have speciation as a thing here. And then I want to show just a couple other things. Here you said, you are the one that is determined to not have an honest conversation about this. You have refused to identify the benefits the mutations conferred to the infected organism. Why? And I just wanted to highlight the honest conversation part of that because I hope my reputation is that, every now and then I get stuff wrong, but I really try hard not to.
Well, Daniel, as I recall, that was a response to you saying I was the one who did not want to have an honest conversation.
Yes, you can see that. I left that in. It's right up. Yeah, it's right up here.
And I do want to have an honest conversation.
Well, to be fair, I said you didn't seem honestly interested in my answers.
Oh, I am. I'm very interested in your answers. I don't agree with them, but I'm interested.
And then the last thing here, I just want to point out, this was not in a conversation with me, but just to kind of contextualize things. There were a couple of examples of this, so I just picked one. But, calling evolution a fairy tale, which I just want to contextualize for everyone, it's kind of where we're at. All right. So now I can take those screens down.
Sure.
So now what I want to do, and folks, we agreed on this format beforehand here, so I'm going to turn it over to Jason for just a few minutes. We got a few slides. I'm going to switch screens because we're doing it from my screen. So this is
Right.
going to take just a minute here.
Right.
So
Right.
bear
Right.
with me because I can only share one thing at a time. So let me pull up the slides. It's this. Okay, and I have to get you back to your first slide now. There we
Sure.
go. Okay. And now we can put this up. Okay, so take, 5 minutes and go through these, and then
Yeah.
we'll
Okay. All right.
chat after.
Okay.
All
All
right,
right.
take
Real
it
quick.
away.
Okay. Sure. Mutation repair mechanisms during meiosis demonstrate that the genome seeks stasis. Well, first of all, just the fact that we're talking about a mechanism here that's called repair. Repair means you've got something going on you don't like, it's bad. And a mutation repair means that we're repairing mutations because there's something we don't want to see. The genome doesn't want to see them. So it's obvious then when the genome is repairing mutations that the genome is seeking not to have changes in the nucleotide sequencing that makes up the genome. So that's the reason the mutations are repaired is that change to the nucleotide sequence, which is the mutation, is viewed as something undesirable. That's why it's repaired. So to me, that's one of the key elements of the logic behind why evolution is not something that would be uh, beneficial to the genome, because the genome itself tries to prevent it by repairing mutations. So I can then just go on. Oh, then well, one other thing is that if you look at online and you would Google, for instance, uh, mutation repair. What's the purpose of mutation repair? You'll get answers like to maintain the data integrity of the genome. Well, data integrity of the genome means we want the genome transferred generation to generation like it has been presented. We want it transferred without changes. That is what data integrity means, not changing the genome as it's transferred generation to generation. And I want to review real quickly, Daniel, an example of why that is so important. And so if you could just go ahead and just go through a couple more slides and get to the YouTube video right there. Now, can you open that link for me
Here
and
we
go to
go. So let me
32?
switch to the video, and then
Yeah,
if you want
and
to,
let me
yeah.
before you do that, let me give you just a little bit of an introduction here to the video. What we're going to see for just 60 seconds, because it's an hour and a half video, but we're going to see Professor William Bielek. He's a biological physicist at Princeton University, and he's lecturing on the human eye, our ability as humans, our eye's ability to detect photons, and what a system, what are the characteristics of that system, what it means when you look at that system and see what it does, what are the implications of how that system works. So we're going to listen again to this professor from Princeton University. Daniel, I'm only asking for about 60 seconds here, starting at I think 32:20, and remember we're getting in the middle of this, but the idea is he's talking about biological systems and then not just vision, but biological systems in life and how perfect they are. Go ahead.
Okay, I just want to make sure. Let me just hang on. I got a bunch of windows open. I got to make sure I got everything set up here because I got your slides. I got these. There's the window we want right there. Just give me a second. Okay, I think we're good. Okay, and I'm just going to full screen that, and
Yeah,
now we're good
that's
to go. Okay,
yeah,
all right.
and
I'm
yeah.
gonna hit play. Here we go. Chat, let me know if you can hear it. Make sure I did this right.
I don't have sound. I'm not hearing any sound.
Let me see here. Hang on. Oh, I can't hear it either. Okay, I did that wrong. Hang on.
Okay.
Yeah, I definitely did it to share the sound, but I'll take it down and we'll try that again.
Okay. Thanks.
Hang on. Stop sharing. Plus, we're gonna do screen share. We're gonna share screen. We're gonna go to this. We're going to go to this, and we are going to share the system sound. We're going to share, and now we're going to do it. Okay, we're going to go back just a second here. Okay, here we go. Try again now, folks.
Thank you, Daniel. Dan.
Good. Oh, wait, you can't see it. Hang on.
Yeah, I can't.
So now I didn't even share the screen. See, here we go. Add to stage. There we go. Okay. Now let me go back a couple seconds, make sure we're good. Okay. Now we're going to do it. Now it should work fine. I've got too many things back and forth. Now we're good. Ready? Here we go.
Driven by temperature, where the molecule is sort of fluctuating and spontaneously does something on its own. There's a certain sense in which you can't do better than this. If you can't count half a photon, because that doesn't make any sense. Your brain can't do any better than is allowed by the amount of dark noise in the detectors. And that's set by these thermal effects that you can't get rid of because we're stuck at our temperature. And so, in a sense that's rather precise, you're operating at the limits of what the laws of physics allow.
Okay, that's good, Daniel, right there. Okay. So that I'm getting an echo. If I'm back on the screen on the slide that he had when he was talking about the vision operating at the limit of what physics allows, the next line on there was life operates at the limits that physics allow. It's not just vision. If you go on to another slide, Daniel, where I talk about the E. coli, go one more. E. coli, the bacteria that Dan and I were discussing, it recognizes chemical signaling at close to the theoretical limits that physics allows. This is also true of other biological systems throughout life operating at the limits that physics allow. Flight is an excellent example. So whether it's the flight of birds or the flight of insects, those flight patterns are absolutely operating at the limit that physics allow. So the point I'd like to make about that is that these things we see today, this operation of biological systems at the limits that physics allow, didn't just start today. The earliest life that existed on Earth would have also operated at the limits that physics allow. What does that mean? That means those genomes, as originally created, are perfect, next to perfect. I think he says in that video where he talks about the eye that it operates, or that system is more perfect than we could imagine. So let's call it perfect. So if you have these genomes which have biological systems which are that beautiful, operate that well, what do you want to do? How do you want that information transferred generation to generation? Well, you don't want to change it because you're going to corrupt it. And that's what these mutation repair mechanisms do. They reverse damage that would enter the genome if those mutations were not repaired, because you have systems that are wonderful the way they are created. They're beautiful and they operate and it's just extraordinary. So you wouldn't want to introduce mutations and try to change that system. And that's why I've said if you have a shrew, the shrew is going to be, it has those systems embedded in its life also. And that shrew will do very well being a shrew, and its descendants will be shrews, not something else. And there's go on to one more slide, please, Daniel. One of the things that mutation, or one of the things that evolution claims, is that we have mutations entering the genomes because that provides diversity. Well, there is diversity within genomes, but it's within the genome as it's created. So diversity is very important, but we get that through the process of homologous recombination. So when gametes are created, the information from the male and the female combine, and it's mixed through a process in the creation of that gamete that ensures there will be plenty of diversification. And an example would be, the male gametes, there might be 250 million or even a billion sperm in a male, but those are all somewhat different. The nucleotides contain a mix of information from the male and the female, so there's tremendous diversity, but it's diversity, Dan, that's created without any mutation. Mutations aren't necessary to create diversity, and the effectiveness is outstanding. We have, I mean, as an example, as you know, I like going back in history, and those ancient humans who created the pyramids, those engineers were really good, and the ability of the genome then to transmit the genetic code faithfully over generations means that 5,000 years later, we have plenty of engineers who can still do great work. And that's because the genome has mechanisms to keep it faithful to the original design. Okay. And next slide, please. So just a summary of these implications is that diversity is actually achieved within genomes via homologous recombination in the gametes. But that diversity, admittedly, that's not evolution, because the nucleotides, the sequencing of the nucleotides is not changing through the process of homologous recombination. And that's also a process that occurs in bacteria also. So there is diversity. It's healthy. It's needed. And some examples, as Dan and I were talking earlier about E. coli. Well, there are millions of strains of E. coli, but there's a single species. It's not recognized. We don't have a million different species of E. coli. We have one. Canis lupus familiaris, that's domestic dogs. 364 breeds. They're all a single species. Everything from teacup poodles to Irish wolfhounds and St. Bernards, but it's still a single species. Going to domestic goats, Capra hirus. There are 500 to 600 breeds of goats, and that diversity is very healthy for the species, but it's still one species. So I would like to make one other point, I think, that proves that stasis is the rule, and that is that as I've talked about the importance of stasis and how genomes tend to maintain stasis, is that means then that in a very real way that many species throughout history have not been able to evolve, right? They don't change. What's the consequence? The consequence of that is when the environment changes, a lot of times species go extinct. And we may not like that, but I mean, I don't know how we can argue that that's not what happens. So we will see this year, maybe a few thousand species or hundreds of species go extinct. And if you look at the number of species that are called endangered, it's thousands. And the reason they're endangered is they're not evolving. They are witnessing stasis in the genome, and that means they stay, even though there's diversity within the genome. They're oftentimes not able to adapt to environmental changes, and they go extinct. So we no longer have a saber-toothed tiger. We don't have a dire wolf. We don't have a woolly mammoth. So that's we see that throughout history. So my last comment on this slide is that I think that stasis that we observe in life, Daniel, means that a shrew is only ever going to be a shrew. It's not going to change over time to become a hippo, an elephant, giraffe, lion, whale, or human. It's going to be a shrew. I'd like to make just one last point, if you would bear with me, Daniel. Next slide. I think this nails it for me, as I've thought about this topic for, 30 years now, is that, or longer than that, about 50 years. A common sense interpretation of the facts suggests that a super intellect has monkeyed with physics as well as chemistry and biology, and there are no blind forces worth speaking about in nature. That's true. It's another way of saying that intelligent design is real because God is real, and God has designed the systems that we have in biology and the universe and the solar system, and we see that in innumerable ways when we just look at the planet and how unique planet Earth is in all of the universe, not just the solar system. But that design says that it didn't happen by chances. As Hoyle says here, a super intellect has monkeyed with physics, chemistry, and biology. Well, that super physics is the creator. That super physicist is the creator. Would you have? I have one. We'll note here that I don't know if you knew this, but Fred Hoyle actually wrote a book about evolution.
I love when people not in biology write books about evolution. That's my favorite
Yeah.
thing.
Well, that's because a lot of people know how to think, not just biologists.
Well, thank you for that presentation.
You're welcome.
Okay, I was taking notes. So I have a few things. So what I do, I got a few things here that I want to point out, but then I want to get into the stasis thing because that's kind of the meat of it.
Sure. Sure.
So if I could just very briefly summarize your argument, make sure that I understand where you're coming from. Kind of the big thing is that there is a tendency or a preference in biology for stasis, for consistency, for maintaining the status quo, and that is contrary to evolution. That works against what evolution needs. That fair?
Yes, that's very fair.
And then the second kind of broader point is that biological systems appear to be designed. They look fine-tuned, engineered, optimal, whatever word you want to use, right? They appear to be, there's like the, yeah, okay. So I want to point out a bunch of small things, but then, like I said, we'll get to the stasis thing because that's like the big one. So first, I have to point out that you were personifying evolution and biology throughout that, which is incorrect because we know the processes that, for example, cause mutations and fix mutations, and they are indifferent. Let's say they are just chemistry and biochemistry, and stepping back more broadly, there's natural selection, and these are not these are not personal processes, right?
All built with information.
That's fine. Just going to ask you, it's my turn, so let me just go through this. Second thing is with things like the eye being able to detect a single photon, for example, or another example you hear about this is like very, very acute senses of smell. And I think intelligent design proponents and creationists in general will put the cart before the horse on this, saying it's right at the level, right at the threshold of where it could detect a photon. And the reason for that is that light is quantized, right? So there is a threshold below which you can be a photoreceptor, but you will never detect anything because you're not sensitive enough to detect the amount of energy that's in play, right? So it's the causality works the other way. It's that selection will not operate on a photoreceptor that cannot detect photon. It's like it goes the other way, right? It's we can show that there are a range of photoreceptors with varying degrees of sensitivities, but they all work at the photon level. You're never going to detect half a photon because half a photon doesn't exist, so you're never going to have selection for such a structure, right? Third thing is that you describe systems as perfect. In biology, there's no such thing as perfect. Perfect is context dependent. What's perfect in one environment is going to be really bad in another environment. For example, we can look at enzymes, where enzymes in humans are perfect for the human body, right? Our enzymes operate at like the optimal physical rate of enzymatic catalysis in humans. But if you were to put that enzyme in a thermophilic bacterium, it's going to denature like that and do nothing. If you put a thermophilic homologue of that same enzyme in a human, it's going to operate way too slowly at 37 degrees Celsius because it's adapted to operate at 70 or 80 degrees Celsius. So perfect is context dependent. And we can see selection operating on that to maximize your, whatever the trait is for your particular context. Fourth, this is a small thing, but it's worth, actually third, my numbering got all messed up. I think I'm up to the fourth thing now. All this talk about trying to not have mutations. The underlying idea there is that mutations are going to be either neutral, which is most of them, or harmful. But we know that there are beneficial mutations, and it's not serious to say otherwise. We know that there are beneficial mutations. We've documented tons of them. And I'd be happy to go through a bunch of examples. Lactase persistence is a good one in humans. You can't assume a perfect genome in the past because, again, there's no, perfect isn't a thing in bio, but also it's begging the question. You said that changes to the sequence are not evolution. That's literally what evolution is. Just changes in the frequency of heritable traits in populations. That's just the definition of evolution.
No, Dan, the sequence when the sequence of nucleotides changes, that is mutation.
Well, it can be mutation or other things, but you said it wasn't evolution, and it is.
No, I think if I go back to that slide, I think I said that the diversity we get from homologous recombination is not evolution.
And I'm saying it is because it's changes in the frequency of inherited traits within a population. That's the definition of evolution. So, like, that very much is evolution.
Are you saying we have evolution when we do not have changes to the sequence of nucleotides? No change in the sequence
in a
of nucleotides.
population. At the population level, if everything stays the same generation to generation, yes, then that population is not evolving. Yes.
Okay. Well, through homologous recombination, there is no change to the DNA sequence. There's change to the information within the DNA sequence.
There's changes to the allele frequencies in the population due to recombination. Yes.
Okay. And that does not change the sequence of the nucleotides.
That's the fun part. It changes the frequency because individuals don't evolve. Populations evolve.
Okay. Well, the frequency of the nucleotide sequence is fine as long as it's not changing. There's not a mutation. So
it doesn't have to mutate. That's the thing. There are other mechanisms beyond mutation that drive evolution. Yes. Yeah.
Drive speciation.
Yes. Yes.
So we could have a species change when we do not have any changes in the nucleotides.
You would have changes at the population level, right, where one population bifurcates into two, and those are going to have different allele frequencies and potentially even lose alleles. But even before you have the speciation, generation to generation, you'll have changes in allele frequencies, and that's evolution.
Would those populations then no longer breed?
Potentially, depending on the situation. Yeah, it depends. We have examples of that, and we'll talk about it. We'll get there. We'll talk about it. Well, because I don't want to get, look, if we're going to get hung up on like the literal definition of evolution, then this is going to be a very tedious conversation. So we'll get there. Okay. The next thing, so sequences changing in any way is evolution. And then the next note I have here is that like speciation happens. This is directly observed. We can like document this happening like right now. And I want to ask, a bunch of people in chat were asking, because it sounds like you're saying speciation doesn't happen. Is it your position that speciation doesn't happen?
Now, my position is that when we say speciation, it can mean so many different things, Dan. There are examples of changes in the nucleotides, the organization or the sequencing of the nucleotides, where two organisms that are 99.5% the same, all of their nucleotides will no longer reproduce.
Right. That can be really similar and not reproductively compatible. Yes. And that would be speciation, right?
Right. Okay.
So speciation happens.
If speciation happens, one of my point is that it's range bound. They're very small changes, and oftentimes when speciation happens, there can be actually a detrimental effect where there's no longer the ability when you reduce your breeding opportunities. That's not necessarily good for survival.
But we
can't have that.
That's a different
speciation. Yeah.
Speciation happens though. Okay, so we don't have to go through that. Okay, most important thing: stasis. Stasis in biological systems. I'll start very simple. Are you familiar with the concept called Hardy-Weinberg equilibrium?
No.
Okay, so this is a thing that happens that I find very amusing.
Okay.
And just for context, what I do here is geared towards non-biologist, non-specialist, right? I try to explain things. My videos are like, here's how to, here's why this creationist argument is wrong. In very simple terms, you don't need to have a degree in biology to understand it, right?
Right.
What I find frustrating when I talk to creationists of all stripes is what just happened here, okay? Because Hardy-Weinberg equilibrium is something that I teach. For context, I teach Bio 1 and Bio 2 in college. First semester, second semester. Most of my class is freshmen bio majors of some kind. In the middle of the first semester of biology, my students learn about this concept called Hardy-Weinberg equilibrium. And it's a concept in population genetics that describes what a population looks like and how that population is operating when it is not evolving. So Hardy-Weinberg equilibrium describes what you're talking about, right? A population that stays the same in terms of its genetics generation after generation, right? That's what this concept, and it's mathematical. There are a couple of equations, and basically if you know some parameters about the population, if that population is not evolving, you can accurately predict what it's going to look like subsequently in various ways, okay? The utility of Hardy-Weinberg equilibrium in evolutionary biology is this. It never happens in nature. It's useful as a null hypothesis, because what we can do is we can look at a population and we can say, if this population is not evolving, there are no changes in allele frequencies from generation to generation, here's what we expect to see. And then we can actually survey that population and we see deviation from the Hardy-Weinberg expectations. And given the nature of that deviation, we can then dig into it and determine what forces are operating on that population that are causing it to evolve, whether it's primarily being driven by mutations being acted upon by natural selection, or it's genetic drift, or maybe there's a lot of inbreeding, which, depending on the context, is often a special case of genetic drift. You can learn a lot about a population by looking at how it deviates from this null hypothesis. And the null hypothesis is that it's not evolving. It's experiencing stasis.
Right.
Okay? The fact that that is the theoretical baseline that never happens in nature, like you can look and you're never going to find a population in nature that's at Hardy-Weinberg equilibrium, that means that stasis is not a thing in biology, like straight up.
Well, okay.
Like, because I'll tell you there's a bunch of things that cause deviation from Hardy-Weinberg, right? And it's actually, we'll do this like I do in class. Do it with your hand, because each finger is a mechanism, right? So you have thumbs up for natural selection, because natural selection increases the frequency of adaptive traits. So you become better adapted over generations. So you got natural selection. Mm-hmm. Your pointer is called gene flow. It's migration. So like you go over there. When individuals move between populations, that changes the allele frequencies. So the populations are evolving. So that's number two. For your middle finger, you do M for mutation, because when mutations happen, that changes the allele frequencies. You can also do it the other way if you want, because mutations can break things. So you could do it the other way. That works too. Ring finger for non-random mating, which is often just genetic drift, but sometimes isn't. So it can be its own thing, and ring finger works well for that. And then pinky finger for genetic drift, because genetic drift is random changes in allele frequencies. And these are most going to have the largest effect on small populations, because you can conceptualize drift as sampling error. So the smaller your sample, the larger the sampling error. So those five mechanisms, some combination of those five mechanisms, are always, always, always operating. Which means,
I can agree with that. Can I ask you?
Just one second. Let me just finish the point, and then yes, go ahead. Because those five mechanisms are always operating, populations are not experiencing stasis. Stasis is not a thing that exists. That would require genetic equilibrium, and that literally doesn't exist in nature. So go ahead.
Okay. So is E. coli evolving?
Yes.
Well, it's okay. All right. So
Yeah.
we can evolve, and we can never change species.
Well, we can change species.
Well,
because something isn't doesn't mean it things can't, right? Because you have different types of selection. So for example, you can have what's called stabilizing selection, which means you're really well adapted to your environment. Like E. coli is really well adapted to human intestines. It's really good at living in human intestines, or
It's good at
mammalian
living in all
intestines more
intestines,
broadly.
right?
Yeah,
It's
mammalian intestines, right?
Yeah.
It's really good at it. So any deviation, right, mutations that cause it to deviate away from the traits that make it really good at that, those individual cells that experience those deviations are less fit than the ones that don't have them. So you have natural selection kind of pushing that population towards that optimum, right? It's a consistent environment and you have it stable. But you can have selection that does the opposite. There's another type of selection called disruptive selection, where you have a population being selected for two extremes at the same time. And you're being selected for conditions A or conditions B, but the intermediate state is suboptimal in both sets of conditions. So that population, instead of being kind of a normal distribution for whatever trait you're looking at, you end up with a bimodal distribution, and that type of selection often drives speciation.
Okay, if we're saying that an E. coli can evolve and always be E. coli,
Sure.
it will always be an E. coli species. I mean,
Maybe. It depends.
That's right. Okay.
We're getting into speculative kind of future evolution,
very,
right?
Very, very, very speculative. That's right. But we can look at E. coli, which has potentially, I've read, as many as 1 million strains.
Sure.
But it's recognized as one
As E.
species.
coli, right? Because
as E.
it's
coli.
within kind of a constellation of sequence and morphological characteristics that allow us to characterize it as E. coli.
Right.
Yep.
Right. So
Yep.
that's what I meant
Right.
by stasis.
Sure.
Okay. So it's not absolutely, there can be no diversity within the species. That's given. There's going to be diversity. Within
See,
the
now it
species.
sounds like you're disputing speciation again, though. And like, do you want to, we can talk about like directly observed examples of speciation. So like, for example, there are these flies called apple maggot flies, where they eat hawthorn berries and they eat apples. And right now we're watching this species bifurcate into two species.
Right.
Right? Like we're watching this happen in real time because the berries and the apples ripen at different times of year. So you have selection to lay your eggs in the two subgroups at different times, which means they're reproductively isolated. So the fact that they live on different plants is a phenomenon called habitat isolation.
Right.
And then you have selection for different reproductive schedules on those two host plants. That leads to a phenomenon called temporal isolation, where they don't reproduce at the same time. So we can actually document right now that though that species, North American apple maggot flies, are in the process of bifurcating into two species of flies that don't reproduce with each other. Like that is a thing we can document right now in the genetics of those flies. We can see mutations accumulating in each of those subgroups that the other group doesn't have, that are causing them to become reproductively incompatible with each other. So you could say like E. coli stays E. coli because all the E. coli that we know of is experiencing stabilizing selection. But there are lots of examples where the opposite is happening, where we can look right now in nature and we can see this bifurcation happening. So stasis is not the rule. It's the case when you have stabilizing selection, but not the case when you have other dynamics at play.
Yeah. I think the point is that the North American apple maggot
If you say
fly
there's still
is-
flies, I swear we're going to start talking about photosynthetic animals and multicellular algae over here. Are you going to say there's still flies? Are you
I'm
going to do it?
going to say the North American apple maggot fly is always going to be a version of a North American maggot apple fly, or North American apple maggot fly. That there's we're not going to be able to step into the future and see it, right? We can't.
No, we don't have to. That's the fun part. We don't have to. We can see it right now. And
You're not seeing it be anything other than a North American apple maggot fly.
No, well, when they become reproductively isolated from each other, which we don't know when that's going to happen, but one of them will no longer be. It'll be a different species. It won't be the North American. That's the thing. Like it won't. It'll literally be something new.
It's going to be a little fly.
It's still okay. So now we're getting into
because it's the practical, because that's what we see, Daniel. We're not the I know where evolutionary theory goes from this. Okay,
I
'Cause I can get we can get changes. So we have two versions of the North American apple maggot fly. So if you repeat this often enough, over time, whales will descend from shrews, and
So what's the limit? So what's the limiting principle in this, right? If
Well, that's a very-
you can
that
have
is-
if you can have genetic changes build up within a species, and you can have one species splits into two species, right? But we can't get whales from shrews. We get whatever, right? You need to have a limiting principle that prevents those types of things from happening. So,
right.
can you articulate a limiting principle?
Well, I think I found an example in bacteria, two bacteria that were, and it's over on let's see if I have it here.
Is bacterial evolution relevant for vertebrate evolution
Well, that's a very good question too. Because a lot of times,
in terms of a limiting factor?
yeah, yeah. I don't know how.
So what's
I don't know how, but
What's the limiting factor that prevents a terrestrial tetrapod from becoming a marine tetrapod? Like mechanistically, what prevents that from happening?
being able to select mutations, which are coding errors. They are that would first the first limit would be the ability of genomes to produce effective errors, which are nucleotide changes that are not authorized, and then for them to
Again,
be-
you're personifying. Authorized is not a thing, though. You're personifying
Well, yeah.
biology.
Well, it's when the genome repairs mutations. So
We'll get back to mutation repair in a few minutes. Don't worry. My point. Let me make my point rather than fishing for it. So let's talk about tetrapods to whales. Whales are tetrapods. Let's talk about this. Terrestrial animals to whales, because, like, for example, you can have primates with longer or shorter arms, right?
sure,
You got dogs with longer or shorter legs, right? You got
all dogs.
right? Yeah, they got you think about something like the maned wolf. It's got those long, spindly legs, right? And then you've got your teeny little dogs that the belly scraping on the ground, right? You look at a whale fin, for example, the forelimb, the pectoral fin. I don't know. That's what it is in fish. I don't know what it is in whales. But the forelimb, it's got the same bones, but the proportions are just different. They're short and fat instead of long and spindly. But they're the same bones. They're the same muscles. It's all the same morphology. Just the proportions are different. And we know that those things can change because we can see variation in that within things that we agree are related from common ancestry. Like, presumably, I don't want to assume your position, but I would assume you agree that all dogs or all canids share common ancestry.
Yes. I think common ancestry to a degree. Although
Sure.
let's say I know that the theory is that dogs have all evolved from wolves.
It doesn't matter. It's just let's just go with dogs and wolves and my point is
there are different species of a dog that all have 78 chromosomes, and they won't reproduce. I think the African dog has 78, and the
you can have the same and not be compatible. Yeah, but my point is there's some level of dog where we agree that they share common ancestry, and they have lots of variation in their morphology in terms of things like the size of their jaws, the relative length of their limbs, the proportions, right? We agree that you can evolve those levels of traits, right?
I think I would say that if you're going to say that the genome itself allows lots of diversity without changing the sequence of the nucleotides, I agree.
Well, it does change, though. We can look at dogs and wolves and maned wolves and coyotes, and we can see that they do have different DNA sequences, right? We can sequence their genomes and see that they're different. So that this does evolve changes in the genome, and we agree that these things share common ancestry, because I feel like, once again, you're kind of walking up to the line of species fixity where speciation doesn't happen.
Well, because what we do is we look at these very small changes between organisms, and then we jump to a huge we jump from there to
You ever see this? You ever see this?
Yeah, I've
You ever
got
see
one.
somebody? You ever see somebody with a little more webbing in their fingers? You ever see
I've
that?
got a little bit. Yeah.
Yeah, there's several genetic conditions actually that are characterized by an increase in the webbing in your fingers. And embryonically, actually, the webbing is there, and what happens during embryonic development is it goes away.
Yeah.
And what happens in aquatic tetrapods is that the webbing doesn't go away, right? You start off with kind of an undifferentiated thing, and then the digits in terrestrial things, the digits, separate from each other. But in marine things, the digits stay kind of together, so all kind of webbed together, right?
Right.
We know exactly what genes are responsible for that. So what prevents the sequence that causes it to go away from changing into the sequence that causes it to not? Because once you change the proportions of the limbs and the webbing of the digits, congratulations, you now live in the ocean.
But there's
Like, it's
there.
not that hard. And
It's
proportions
hard.
of body fat, like for insulation, that's different people have different proportions of body fat inherently. Like, we're talking as though whales and their terrestrial ancestors are like so incredibly different from each other. It's the proportions of a couple significant traits that are relevant for the different environments are what's changing.
Yeah.
And it's
The,
just sequence differences. And we agree
oh,
that mutations happen.
you, there is no way.
Yeah, I mean,
for
this is the same thing.
genome.
Bat wings are the same. It's fingers with the webbing. It's the same. Yeah.
The point is there is no way that to sequence mutations in the right order to create complex organisms. There's so if I
Let's
have
do this one.
oh yeah, let's do this one. So let's take the human eye. Or
Yeah,
any eye.
you need a certain set of mutations in the correct order, and if you don't get them in the right order, and the probability is like 1 × 10 to the negative bajillionth power. Yeah, I know.
You've
that
just become a
assume-
creationist.
Here's why that's wrong, because it assumes that there is one genetic pathway and one pathway only to each trait, and that is demonstrably false. That is why that is wrong. This comes primarily from a 2004 paper by Douglas Axe from the Discovery Institute, and it's wrong because he is aiming for a specific target polypeptide rather than looking for a function. And what we find when we do the assay to say what sequences will do a particular function, it turns out tons of sequences will do the same function. There's a paper from I want to say it was 2019, but don't quote me on it, chat. Someone's probably going to remember it because you'll remember the paper. But what they did is they took the lac promoter in E. coli, and it's about 100 nucleotides long, and it regulates gene expression of the lac operon in E. coli. And what they did is they replaced those 100 nucleotides with 100 random nucleotides. And they did this like it was thousands and thousands and thousands of random 100 nucleotides. Tons of them functioned as the lac promoter. Tons of them. And then they did a really cool thing. They let the functional ones continue to evolve. And within one mutational step of those randomized sequences, they found promoters that operate more efficiently than the wild type promoter. So this idea that the probability of a certain structure or certain trait evolving because the target sequence is so rare that it would be more than there are atoms in the universe, right?
Exactly.
That's
You're right.
nonsense because there's more
that's
than
not
one
nonsense.
functional sequence. That's the answer. And you can say, no, there isn't, but literally this paper found dozens and dozens and dozens of random sequences that functioned as the E. coli lac promoter.
Okay. When you can't go into nature, you just can't see that in nature. We'll never see a sequence of mutations that are generated randomly that are then selected by nature, which over time create an eye. So it's a theory when you say, oh, that could happen. Or the
I
land
can
animal
literally
could be
tell
a
you the
whale,
mutations
or
that do that. I can literally tell you the mutations that do that. It's a receptor in a cell called a G protein coupled receptor. It's a subset of proteins called serpentine proteins because they zigzag back and forth across a plasma membrane. Now, G protein coupled receptors are chemoreceptors. But there's a mutation that can occur that causes them to, and basically what happens is outside the cell, a chemical like a hormone binds to it. And then that causes a structural change inside the cell, which activates another protein and it signals something inside the cell. That's how a G protein coupled receptor works. There's a mutation that can occur in G protein coupled receptors that causes them to be sensitive to photons. So when a photon hits it, it causes that change inside the cell. Boom, now you have a photoreceptor.
Okay. You can't, everything you've just said is a way to explain something that we never get to see. We never get to see an eye created piece by piece. We never get to see nature select all the mutations that it would take to create an eye over, or
Do you
over between the time that a cell was a cell sensed light to the point where it gave it an advantage because it had an
Well, you don't have to. It
Well,
just happens
but-
by chance. That's the fun part. You don't have to try. It just happens because mutations just happen.
yeah.
So here's a question for you. Do you think multicellularity could evolve? Do you think it's possible to go from a unicellular organism to a multicellular organism? Do you think that's something that could happen?
And can I explain why not?
Please
no, I
explain
don't.
why this thing can't happen. I can't wait to hear why it can't happen.
Sure, because we have several experiments where from algae and from yeast, the yeast and the algae were stressed, and what happened was the single cells took on separate functions to allow the colony of yeast to survive, and those single cells that perform different functions then sometimes they would communicate, and they would then be able to take on different functions to help the colony survive. Okay, and then so that's an example of
Question for you for that. In the yeast and the algae, were those cells, did they like stick together? Were they separate cells that stuck together, or was it a single cell and then it divided clonally into the larger structure?
I'm not sure, but I think it's single cells that stuck together.
It's single cells that, well, you kind of mix the two things there. Is it so? Did they team up, or did they start as one and then split?
They teamed up. I believe they teamed up.
Wrong.
Oh, okay. All
That's
right.
the ancestral state. Is that they have facultative colonialism, which is a response to predation.
Okay,
because
so
the clumps were too big for the predator to ingest. Right. That's the ancestral state. And I'm talking about the yeast specifically. This is the chlamydomonas.
Okay, maybe I did get it wrong, but the
Well,
point
hang on.
is
Let me
that-
finish the explanation here,
Okay,
because
all right,
because
right.
so the ancestral state is that they would become adhesive to each other. In the presence of the predator, they'd stick together. Predator goes away, they would go back to being unicellular. That's the ancestral state.
Right.
They also they've documented, not something that you see typically, but you can see mutations in these algae where the cell wall would fail to divide. So you would have one cell, it would go to split into two, but they'd get stuck together because the cell wall wouldn't divide, right? So those are kind of your two ancestral options. But in this experiment, where they grew these yeast for 2 years in the presence of the predator,
Right.
they found a third thing, something that was new, where the cells would divide completely, including the cell wall, but then they would stay together due to the presence of an extracellular matrix, right? And that would lead to multicellular structures that were up to, I think they got up to 32 structures, but 32 cells, but the most common was 8 or 16 was kind of the optimal size. And here's the really fun part of this. You only had in that multicellular structure, some of the cells would reproduce and other cells wouldn't. So you had somatic germline differentiation. Do you know what those words mean?
Somatic cells are cells that in the human body they're not the gametes. They're
You got
cells
it. Yeah,
that
yeah. So the somatic cells are everything except the germline that make the gametes. The germline is what makes the gametes. Exactly. And here's the thing. If you have somatic germline differentiation, that is the calling card for true multicellularity, because it means that that group of cells is satisfying a concept in population genetics called Hamilton's rule. And that only works Basically, it's a concept that explains altruistic behavior, and multicellularity is the ultimate altruistic behavior because your skin cells, your muscle cells, they're never going to reproduce, but they're helping your germline reproduce. And that only works when they're genetically identical to each other. So the fact that you have in these algae multicellular structures that are clonal, right? They're not aggregate, they're clonal. So they're from a single cell, so they're all genetically identical, and they have germline somatic differentiation because not every cell is reproducing. And then they remove the predator. Because remember, this colonial behavior, this aggregate behavior that they showed, that was adaptive, it was facultative, so they could do it and undo it. With this new multicellular cellular trait, they removed the predator, and it stayed multicellular for 2 years. The whole duration of the experiment. It was permanent. So it was true de novo multicellularity, permanent with germline somatic differentiation, evolved in the lab in the span of 2 years, and then persistent for another 2 years. And that was as long as they did the experiment. They kept going, but it was 4 years.
Yeah. Can I point out, Daniel, am I not right in saying that the single cells always reproduced as single cells? I mean, as single cell.
No, that's not what happened. That's the point.
So you had a group of yeast, single cells yeast.
Well, these were algae. The yeast experiment was different. We can talk about that too if you want. That was a different thing.
Yeah. All right. Well, I think at the end of the day, at the end of the day, don't we have yeast that are only single cell yeast? We don't have.
Sure.
We don't in nature. There's yeast, and there's not. There's no yeast variation where there's a cell, a creature.
To my knowledge, there's no multicellular yeast, but we can experimentally evolve multicellular yeast. It's called snowflake yeast. Yeah. And similar to the algae, it's permanent and it has cell specialization. So it is true multicellularity.
I think the multicellular. I think there are multicellular algae.
There are, but this lineage was not it.
Yeah.
Yeah, the Chlamydomonas only has unicellularity in its evolutionary history. It has no history of multicellularity. There's other types of yeast. So Chlamydomonas is a group called I want to make sure I don't get it backwards, they're chlorophytes. The multicellular green algae are mostly charophytes. That's the group that's closer to plants. But Chlamydomonas is not in that group. It's not one of the filamentous ones, or it's not something complex like Volvox or something. It's a unicellular algae that has no multicellularity in its evolutionary history.
And I think when we look at nature, go out to a pond and you look at the bacteria. If you look at yeast and you look at yeast and single cell algae, and look at them in nature, generation after generation, they're going to be single cells. So
that's fine because stabilizing selection. Can multicellular algae evolve? The answer is yes, right? Because we can watch it happen in the lab.
It's not evolving in nature. So
okay, so now it's not just we can't see this thing happen. It's we can't see this thing happen in real time in nature, right? That's the criteria.
Nature's where algae lives. Nature's where bacteria live.
So
you take Richard Lenski's experiment with
E. coli,
E. coli, and he's got some cool things happening with citrate in the lab. And if those E. coli were placed into nature, they would
because
die.
They've been selected for the lab environment. Yes, that's how that kind of goes back to what I said earlier, that nothing is perfect in evolution. It's always context dependent. Now, I want to step back and first I'll point out that we just went through 2 experiments where multicellularity evolved, and your conclusion from that was, well, multicellularity still can't evolve, which you do you. But I want to go back to this stasis thing because we talked about how Hardy-Weinberg, which I will remind everybody, foundational concept in biology that you learned about half an hour ago. So just want to that's where we're at. In addition to the fact that Hardy-Weinberg equilibrium does not happen in nature, which right away shows that stasis is not a thing, we can actually directly document selective pressures that cause more variation rather than suppress variation. And I want to just establish up front, if we could establish something that is adaptive, and we could show it's adaptive because we can measure the strength of selection, if we could establish that such a mechanism to increase variation, increase the number of mutations is adaptive, then I feel like that kind of contradicts the concept that stasis is the order of the day, right?
The issue is that
Well, what do you think? If we could identify a trait that increases mutations or increases variation in some way, and it is adaptive, then that contradicts the idea that stasis is the general rule.
Daniel, with the ability of genomes, all genomes, to identify changes in nucleotide sequencing when it happens and target it for repair, then you're talking about a situation where am I not right in saying that evolution then is going to succeed when the repair mechanisms that evolution created don't work? When the repair mechanisms are not able to repair a change in nucleotides that
Did you know that there are what are called error-prone repair mechanisms, where the repair causes more mutations to occur?
I did not know that.
Yeah, that is a thing that you can document. And here's the really fun part. If you remove or suppress the error-prone DNA repair, the cells or the populations that don't have it, which is to say have a greater degree of stasis, they lose out to the ones that have it. It's more fit to have the error-prone repair than to not have it. Like it's the thing, for example, in bacteria, since you like to talk about bacteria, there's a thing that bacteria have called SOS repair. It's a last resort where the cell is in an environment that's it's where the cell is not well adapted. Maybe it's high salinity or the pH is wrong or something. And the cell will basically engage in this thing called SOS DNA repair, which causes more mutations. And in doing so, it allows the members of that population to sequence more, to sample more of sequence space, which is to say you try out lots of different things. Because in this new inhospitable environment, you're not well adapted to it. You need to find something that works. You need to find a new genotype. So you have this error-prone DNA repair that allows you to find beneficial mutations.
Is that a bacteria?
Yes.
Yeah. Their bacteria have a wonderful ability to share information so that they become more adapted to their environment.
But it's not recombination though. It's individual cells basically engaging a mechanism that causes more mutations based on the environment.
Right.
So they're not like communicating with each other or doing recombination or anything. It's like a cell under harsh conditions, these mechanisms switch on.
And does the cell then, when it creates those additional variations, does the genome have instructions in it that tell the bacteria to generate the mutations? Basically,
it can increase the rate of mutations. It cannot pick which specific mutations.
Right. So that's a information included in the cell, which enables the cell to, at the end of the day, that bacteria is going to be that same species of bacteria. It doesn't change species. It enables it to survive. It becomes
changing the topic. With respect, you are changing the topic because the question was, your central claim is that stasis is the kind of trend, the order of the day, the thing, right? Everything tries to maintain, keep it the same. We literally have mechanisms that are adaptive, and we can directly show that they're adaptive, that cause more mutations to occur.
Okay,
and it's not just bacteria. If you don't want to use bacteria, we could talk about trypanosomes, how they undergo very rapid recombination to evade your immune system. We could talk about how viruses prevent their host cells from repairing the viral DNA. Like, we have lots of examples of this where we have specific traits that are adaptive that increase the mutation rate rather than suppress it.
Okay. And in those cases, what we're doing is, if the genome of that organism actually, in that situation, causes more mutations to occur, what that organism is doing it's finding a way to always be that organism. So if it's going to be a more successful bacteria, it's going to stay that species. It doesn't change species. It adapts to its environment because it has instructions in the genome which allow it to adapt to its environments. It's like the E. coli. The E. coli has millions of variations, but it's still E. coli.
So just to be clear,
So
I understand you.
So let me ask. Can I ask? Does the
Please,
bacteria,
go ahead.
Does the bacteria, after it generates many more mutations, does it go through speciation then and become a different bacteria?
It depends. It depends on the answer in biology for a question like that is always it depends, right? And because I'll give you an example. If that bacteria evolved for you've got a swamps, right? You've got like a marshy, swampy system, and for some reason, one of the waterways in that environment, full of different bacteria, one of the waterways in that environment becomes more acidic, right? Well, now the bacteria in that one, they engage SOS DNA repair. They get lucky. They find mutations that make them tolerant of that more acidic pH, right? So now you've got a subgroup of those bacteria that are now can only exist in the more acidic waterways, but they can't exist in more neutral ones, right? Those are now going to be reproductively isolated and genetically isolated from the rest of the bacteria. So like that would be an example of speciation. Does that always happen? No, of course not. Could it happen? Yes. Now here's my question for you with this. How is the thing that you said logically connected to the previous thing we were talking about, which is that genome suppress variation? And I said, here's a bunch of mechanisms where they actually cause more variation. And you said, well, would that be speciation? How is would that be speciation logically connected to they suppress variation when they actually don't? Like, I think connect the dots in that argument for me.
We are talking about the ability of bacteria to engage their environment and either with gene transfers between bacteria,
Now or other mechanisms.
Or other mechanisms. Even if it's the ability to generate genetic change within the bacteria to make it adapt to environments that it could not previously adapt to, that is an adaptation that enables that bacteria to function as that bacteria.
And what I'm saying is connect the dots to the genomic stasis for me, because your central argument was DNA repair causes stasis, right? You suppress mutations and keep it the same. And I'm giving you multiple mechanisms of the opposite happening. Okay. And you're saying, but it's still a bacterium. That doesn't matter.
Oh, yes, it does.
For the argument that you're making. Your argument is, so to be clear, we're giving up on genomes are geared towards stability and nothing else. We're giving that one up.
No, I'm not.
Okay, then stick with that then. What's the counter argument to error-prone DNA repair? What's the counter argument to bacteriophages actively being adapted to evade the DNA repair of their host? Which, here's a fun thing you can do, because I've done this with these bacteriophages. If you chemically mutagenize them, you will see selection for them to start utilizing their host DNA repair mechanisms to keep the mutation rate where it should be, right? So if you artificially increase the mutation rate, they'll bring it back down. If you artificially decrease the mutation rate, which is what you say should be happening, right? That should be better, according to you. They will adapt to bring it back up because that's a more fit state to be in.
Okay. All right.
Like, that's my point. Okay. And whether it's speciation or not doesn't matter, right? The point is that you're wrong about the overall idea being to suppress variation. Like, empirically, that is wrong. And I want to be clear about that. And then we could talk about speciation. Is it still E. coli? Whatever. But I want to be clear about the central thing that, the thing that we talked about in your email. You said the telos of mutation repair, right? Like, that central thing is deeply fundamentally wrong.
Okay.
That's my
point.
And
Daniel, I want to go back to the point you made earlier that can we look at bacteria and say that what happens bacteria is functional for mammals?
Yeah,
okay, so
we can.
Well,
same selective pressures operate, same 5 things to violate Hardy-Weinberg. Yeah, it is relevant.
I think that what you do with a bacteria and a bacteria's ability to adapt to its environment by possibly even increasing mutations is going to give that bacteria a very good advantage, and it will help that bacteria survive. But that's a
All
mechanism.
right, thank you for acknowledging that. I want to just say before we move on, I just want to say thank you for acknowledging that, because that is like our central disagreement coming into this. And if you agree that it's beneficial in some cases to increase the mutation rate, that's an adaptive trait, then that's
bacteria.
great. Well, we also see it in eukaryotes plenty. You see it in trypanosomes, for example,
All
right?
sure,
They have a similar thing.
And in humans, what evidence do you see that in humans it is good to have an increase in mutations in the gamete cells? Which gamete cells?
The genes for the immune system, you do want to have a greater rate. It's intragenic recombination. You want to have an elevated rate because that allows you to target more pathogens. Yeah,
That's right, and that's designed. That is
that's just
designed-
asserting that it's designed. You can't actually support that empirically.
Oh, it's very much designed, because we're
Who
born
did the
with an immune
designing?
system.
Who did the designing? What mechanism did they use? Like what? Because if you ask me through what mechanism did this trait evolve, I can either give you an answer, or I can say for that specific trait we don't know, but here's an example of that kind of thing that would evolve, even if for a particular example we haven't figured out the answer, right? Now, if I ask you, you're saying, oh, it's clearly designed. We know it's designed. Through what mechanism was it designed?
By the, I'm going to go back to Hoyle's Fred Hoyle's statement at the very beginning that it's obvious when you look at the complexity of the
Not to me.
yeah, I know it's not obvious to you, but when you start talking about the immune system and its ability, and it does generate many different solutions by creating potentially billions of different proteins to figure out which one is necessary to fight an infection. Okay, and it does that randomly till it finds till it
Right.
finds
It's
the right
right.
one.
It's a probabilistic process. Exactly.
Right,
It
that's right,
generates
that's right.
lots of variance. Right. That's how evolution works.
But there's information that causes that system to do that. That doesn't happen
It's
because
just genes. It's just biochemistry. It's not
genes are
information
made-
like a computer.
Oh,
it's
it's
not
very-
information like a
Oh,
book.
very-
It's
very much-
literally
very
just
much like
chemical
information.
interactions.
Well, you're disagreeing now with Richard Dawkins, who says that the genome is very much like.
I love it when people say that as though I have to like kneel at the throne of
Well,
Richard Dawkins.
okay,
I disagree
okay.
with him on a lot of stuff. That's what like scientists do. We disagree with each other. All right, here's the thing. You agree at the beginning. You said you accept the ancient age for the Earth, right? You're old Earther, not a young Earther, which means that I'm gonna presume, and correct me if I'm wrong, I'm gonna presume that you agree that like the biodiversity that exists now didn't always exist in the past. You kind of basically accept that there have been changes in biodiversity over long periods of time. Is that fair?
Yeah, that's true.
Okay. Now we know, because duh, that the things that like as biodiversity changes, the genomes have to change, right? Like we know that happened. We know that genomes have to change. So it seems like
we don't. Wait a minute. I'm not agreeing that genomes have to change. I think the mutation repair mechanisms are a mechanism to cause genomes not to change.
well, we just talked about how that's not universally the case, and genomes do change because Hardy-Weinberg isn't a thing that happens in nature. So like we covered that already.
Well, your heart that you're conflating any difference in information in the genome that occurs without having a change in nucleotides with evolution.
I would ask that you not tell me I'm wrong about a concept I explained to you an hour ago. I think that's a fair request.
Well, okay,
So here's
that's fine for
the
you to
point.
request.
Here's the point I'm making with the ancient Earth thing. If we agree, right, that the Earth is old, biochemistry or biodiversity in the past was different from the biodiversity now. We agree on that, I think.
I think there are animals that used to live on the Earth that don't live on the Earth
And
today.
there are animals
So
that live
that's-
now that didn't used to live, right? It
Yeah,
works in both
right,
directions.
Right. And there are
Okay.
animals that have been there, ancient animals like bats and dragonflies.
Yeah, I mean, crocodiles, coelacanths, we could find that stuff very similar going back hundreds of millions of years on the fossil record, right? Yeah, that's fine. My point is this: to get from the stuff that was there to the stuff that's now here, right? To get
Yeah.
from point A to point B, you have to have genomic changes. Just say yes and we can move on. This is the easiest question of the night. Like, yes, obviously you need to have genomic changes. So my question for you is if those genomic changes didn't occur through evolutionary processes,
Yeah.
how did they happen?
Well, that would be an answer that anybody who understands or believes that there is a intelligent designer.
So they occurred through design. What,
They have to be designed from the beginning.
through what
But
mechanism?
the power of the creator. There is a, I cannot identify a mechanism.
Okay.
And but that doesn't mean that when you look at the design that we don't see design. Yeah, design. When you get to Dan, when you get to systems that operate at a level that is the limit of what physics allows, that is absolutely proof of design. It's absolutely proof
I am going
of the
to
intelligence of
ask
the designer.
I'm going to ask you a question that I know the answer to, but I'm going to ask anyway, because it's a really important experiment when it comes to the nature of mutations and how mutations
Right.
occur.
Right.
Because what you're saying is either, it has to be one of two things. Either the set of genetic changes, the set of mutations that happened over time, either had to be preordained or the designer had to be actively causing each specific mutation to happen in the right order. It has to be one of those two things.
No, it doesn't have to be, because if there's a creator that is omniscient, omnipotent, omnipresent, then just as the universe was created, life could be created. I think that the genomes are not something that change in large part due to mutations. I think it's obvious that mutation repairs
show that.
Well,
So again,
in
the repair
fact,
thing, we already addressed the repair thing because like there's
fact,
error-prone repair, there's other mechanisms that promote variation. Like that is, yeah, I give you another one because it's fun. I'll just give you another one because I love this stuff. When you have host-parasite relationships, you get a dynamic where the more variation makes you more competitive. So what you see is that in species, the one I'm thinking of is snails, for example. There's I think it's in New Zealand, where they can reproduce sexually or asexually. And the frequency of males in a population of these snails is proportional to the parasite load. So the more parasites there are, that's imposing a selective pressure for sexual reproduction. And as you put in your slides, sexual reproduction promotes variation. So we have another mechanism that's operating against stasis. And not only do we just have the mechanism go, aha, not stasis, we have a specific evolutionary pressure that we can document that's driving it, right? We have the whole picture. So like there's another example. So you keep going back to this DNA repair keeps things the same. And I can just keep giving you examples where that's not the case.
Daniel, will you give me examples of the human genome other than the designed immune system that is generating? So where else do we see in humans a preference for mutations?
The EPAS1 allele in Tibetan populations. That's a mutation that is beneficial living at high altitudes.
Okay. All right. And
lactase persistence. There's at least 6, but there's 2 that are most common, and they all occur in an enhancer for the lactase gene, and it allows you to drink milk throughout your life. I'm sure you knew that. And the 2 main alleles, one evolved in Scandinavia, the other in North Africa, probably like modern Morocco. But this happened at like the dawn of agriculture. And because either it was too hot or it was too cold, you had crop failures. And if you could drink the milk of your livestock, you survived longer. So, I mean, you can have selection for new variation. That is like a thing. We can document that. That's an easy question.
all right. And when we do that, we are going to be playing on the fringes of the human genome. I'm not going to argue that being able to digest milk is not a good thing. That's a good thing.
All
billions
right,
of people.
let's
There's
keep it
billions.
rolling then, because now we talked about multicellular yeast, multicellular algae. Like, you need a limiting principle. If you're going to say, well, it's still a human, it's still a fly, whatever, then you need a limiting principle. And if we're getting to the point where we can
Well,
show
Daniel,
the
don't we see the
evolution of
limit?
multicellularity,
Don't
where
we?
we can show animals becoming photosynthetic, we can show, what's another good one? We can show completely new genes from previously non-coding DNA. Like, you need a limiting principle that prevents A, not just from turning into B, but from C, D, E, F, G, all the way to Z, right? And if we can show, like, literally, we can watch these things happening. We can show that these new traits are evolving in real time. We can witness it. That limiting principle vanishes.
No, it
whatever
doesn't vanish at
was.
all. It doesn't vanish at all, because what you're doing is you're enabling that organism to better adapt to its environment as that organism. So that organism isn't ever going to be anything
So
but that type of organism. That
is
bacteria, a bacteria will always. So it's I don't know if it's, I don't know how we can put a defining boundary to it, but the boundary
Exactly.
exists.
That's my point.
Okay,
That's
but the boundary
exactly my
exists.
point. Because if
Okay.
you're going to say
Is a
that
bacteria,
an animal,
Daniel,
right?
is a bacteria ever going to be anything other than a bacteria?
Yeah, okay. Well, we have animals that are photosynthetic. Like, we have animals that are right now going through the process of endosymbiosis, right? And if you're going to tell me that sea slug, oh, it's still just a sea slug, it literally doesn't have to eat for a year because it's photosynthetic. It's an animal, but it is photoautotroph. You're going to tell me that's just a sea slug. Is
Daniel,
that
it's just a sea slug. Okay.
Okay, great. Then if your position is that it doesn't, multicellularity evolving, animals becoming photosynthetic, that doesn't count. That's still the same lane of organism.
Is it not?
Then
Is it not the same lane of organism?
you tell me, do you think
It
that an
absolutely
animal
is the same
becoming
lane of organism.
photosynthetic is like,
A sea slug.
a
is
different
sea slug,
family,
yes.
for
It is.
example?
It's going to be a sea slug.
It's going to be within the clade,
right?
right? It's going to be in the clade because that's how cladistics works, the law of monophyly. It's always going to be within that clade. So it's in genus Elysia. It's always going to be in genus Elysia. But do you think those are arbitrary ranks. Like, the ranks don't matter. What matters is the biology. And
The biology very much matters.
and if
those
something can
boundaries,
go from being an obligate heterotroph to a photoautotroph, and it does not need to eat, if something can make that transition, right? If an organism can make that transition, and we can witness it happening, which is the case, then I struggle
why
to
can't
see
it
where the
prove-
limits are.
because what you're doing is then you're assuming, because that small change you saw for that
That's a
organism-
small change. Okay.
Right.
Okay.
Okay. But you're going
Okay.
you're going to see that amount of
That's
change,
fine.
and then you're going to extrapolate that to, oh my gosh, that's how shrews.
What
That's
is
how
a
whales,
larger
that's how-
okay, here's a question
That's how
for you.
whales. That's how over 200.
No, here's a question for you. That's a small change. All right. I got a question for you, and then I think we can kind of put a bow on this because we've been going for about 90 minutes, and then we got some questions from the audience here. But here's the thing: so these things we can witness, right? We can witness new genes forming from non-coding DNA, animals becoming photosynthetic. There's literally dozens of examples. Multicellularity evolving. We can see reptiles evolving live birth, so they're changing their reproductive mode. Now, these changes that we can witness, these are all small changes. These are just changes. If you were young Earth creationists, you'd say they were within kinds, but you're not. But that was, right? These are small changes. What are the big changes? Because I will tell you, changing your reproductive mode or becoming multicellular is a way bigger change than a terrestrial tetrapod becoming a whale, because you don't need any new parts to do that.
You need lots of new parts to go.
No, name them. Name the new parts.
Oh, okay. The whale has to have the
Blowhole. Yeah.
on the
Do
top
you
of the
have
head, right?
nostrils? Yeah. Do you have nostrils?
Yeah, and
Yeah,
they're not
do
migrating
hip-
to the top of my head.
Where do hippos have their nostrils? They're sticking up the top, right? It's the same structure. It's just in a different place.
But
It's
you're
the same
assuming it can
structure.
move, Daniel. It
How
never
do they
moves.
move?
Yeah, I think if you watch
Yeah.
hippos, if you watch hippos being born, those nostrils are going to be at the same place on every generation.
How do they
of hippos
move? Yeah,
you're
they
ever going to
move
see.
due to changes in gene expression during development because it's basically it's where the tubes develop. It's just instead of developing over here, they develop over there. It's just where does that tissue develop during development? It's
Okay.
the same genes. It's the same features. It's just where in the embryo does it develop? That's it.
Okay.
It's all because the question was, what's the new structure? And you said a blowhole. That's just nostrils. It's not new. It's in a different location. So what's the new structure? That
Oh,
whales,
it's definitely the blowhole, because the blowhole's not the
those are
blowhole-
nostrils. That's just the nostrils. That's not new. It's just in a different place. The answer is the involucrum. It's the ear bone. The answer is the thicker ear bone is actually new. But we see that in some terrestrial vertebrates and fossils. So
Yeah.
it's not actually new.
Yeah.
Yeah, it's just a thicker bone, and we can change the thickness of bones by stopping the expression of genes that promote bone growth later in development. The bone gets thicker, right? It's like I said, going from being a heterotroph to a photoautotroph, or going from unicellular to multicellular, is a much bigger transition than going from a hippo to a whale. Because again, hippos and whales, that's the same body plan. Those are the same parts. The proportions are different. But going from unicellular to multicellular, you need new parts. Going from laying eggs to giving live birth, you need new physiological features that enable you to do that, right? And those are changes we've witnessed. So if you can witness the changes that require new parts, obviously the changes that don't require new parts.
We've never witnessed an animal go into the ocean, a land animal, go to the ocean, start spending more time in the ocean, and then become adapted to the ocean, lose its legs.
Individuals don't evolve. It's not how evolution happens. It would take multiple generations. And we actually do see this in certain species. Oh, what's the species of wolf? Is Dr. Chris Thompson, are you still here? What's the wolf with the webbed paws? Because we actually do see a wolf that is primarily an aquatic hunter. I forget the name of it. But Dr. Thompson, he was here earlier, and if he's still here, he could tell me what it is because he's brought it up a bunch of times. But yeah, there's wolves that are actually like, we can see this happening. They primarily fish almost like bears, and they've more webbed paws than all the other wolves. And it's like, yes, this is a thing we can document. They're adapting to being aquatic rather than terrestrial hunters. Yeah, that's literally a thing. Yeah.
Yeah. Okay. So it's literally a thing, but we don't have an animal that is on the land going to the ocean, evolving. You know, these gaps at one time there were transitions for all of these creatures, and those have all disappeared. So now we just have land animals, and we have whales, and we know that
do lungfish exist?
Lungfish exist, yes.
Lungfish exist. What about mud skippers?
Mud skippers exist.
How about seals?
Seals and otters exist.
Yeah.
Yeah.
They kind of spend time in both places, right?
Yeah, they spend time in both places.
They could dive really deep. They can hunt like whales, but they don't have a blowhole. They have traditional nose. Yeah. Just saying.
Yeah, okay, I understand. They also their progeny are also never changing, or their progeny generation after generation. All we see is the otters give birth to otters. There's no reason to think that the otter's ever going to be anything than an otter.
And yet we can evolve multicellularity. We see lizards. You could say, lizards always give birth to lizards. You could say lizards always give birth to lizards, right? The fact that they give birth makes them, it's new. Because lizards lay eggs. And we can document this happening. There literally was a lizard where she laid 2 eggs and then gave birth to a live young in a single clutch. There are lizard populations that, since we've been watching them, have transitioned from primarily egg laying to primarily live birth.
A lizard will be a lizard.
It's still a lizard, but that requires more difference than a hippo becoming a whale, because you need new mechanisms. You need new biochemical interactions. It's not just changing the arrangement of structures that you already have.
Yeah. Well, the degree of structures that have to happen to allow a land animal to develop into a whale and then
It's all the same.
dive. Well, yeah. Is it understandable that a hippopotamus would be able to, or an animal that descends from or is descended from a hippopotamus or some land mammal would be able to dive, what is it, 6,000 meters?
Eventually,
5,000
I mean,
meters?
eventually, yeah, if it was driven to do so by a scarcity of food on land and a lack of competition in the oceans. Yes. Yeah, it's a concept in ecology called the competition dispersal trade-off. There's a thing called resource partitioning or niche partitioning that, again, drives things to adapt to new resources.
Yeah.
So you can
And-
select for incrementally new traits,
Okay,
slightly
Daniel-
shorter and more webbed arms, right? Slightly higher position nostrils. You can select for these traits generation after generation. And the thing is, if you can select for a slightly shorter, slightly wider limb, which I think we agree we can do because we can see that happening within a few generations, what's to stop you from continuing that trend as you move more and more towards the ocean? The answer is there isn't a mechanism that causes it to stop.
Oh, I think there is. I think the environment, we can look at environmental pressure. When animals face environmental pressure, how do they respond to it? You know how animals are going to respond to it on the earth today when we look at nature? Nature is a very good laboratory. The way nature is
Exactly.
going to,
That's
the way-
evolution. That's how evolution works. You just try stuff. Yes.
Yeah. And so evolution works. Why will several thousand species go extinct this year? Because they
Because
don't
their
evolve, right?
environment is changing faster than they can adapt to it, or their environment is going away. One or the other. Yeah.
Right.
Right.
And we see that
Yes.
same thing in the fossil record where the environment change and
Over longer periods of time, but yes, it's slower in the fossil record, but yes.
Okay.
Yeah.
All right. So I'm seeing an inability of organisms to adapt to their environment when the environment changes. The environmental pressure causes them to go extinct. It doesn't work on them slowly, so that over time the hippo can spend millions of years.
I mean, it literally
evolving
does
to
though,
become-
because the hippo example is not different in principle from the apple maggot fly example. It's you have one resource where there's a lot of competition for, whether it's hawthorn berries or prey on land, right? And you have another resource where there's less competition for, whether it's apples or prey in the water. And the concept is the competition dispersal trade-off, where if the cost of dispersal of using the suboptimal resource is lower than the cost of the competition, because even when you win the competition, there's a cost to it. If the cost of the dispersal is lower, then that is selected for, and you get what's called directional selection. And over time, you can get a lot of differences in directional selection. There's an experiment that spanned it was about an 80-year experiment looking at, I think it was oil in corn, where the original range of oil in a corn kernel was like 4 to 6%, and they were able to evolve a line with 0% and a line with I think it got up to 80%, and well outside that original range, which illustrates the degree to which there is no limiting principle. The existing range of phenotype does not constrain the possible phenotype. Your nostrils being on the front of your face does not constrain them being on top of your head 1,000 generations from now, right? That's the idea.
It's a fairy tale. There's never going to be there's never going to be, there's not going to be.
It's a great place to end it.
Nostrils.
Who are you going to trust?
No.
Who are you going to trust, Jason or your lion eyes, right? All right.
No,
that's
I'm
a
not
great
gonna-
that's a great way to end it. Thank you.
No,
I couldn't sum it up better myself. Let's go to the questions from the
Daniel,
chat.
Daniel, before we go, there's a principle in biology called and earth science called uniformitarianism, and uniformitarianism says we cannot look at what we see today and say that this is how it operates today, but if we go back in the past, it would have been different. So today, what we see are
We don't do
organisms,
that anymore.
organisms that are not able to adapt to the
In-
environment, they become extinct.
Okay.
Now you're going to go back and say, okay, if I
I'm
go
not a
back 100 million
geologist.
years, if I go back 100 million years, we're going to see that this operates different. The organisms, if we just go back in time, organisms will
I'm
be able to
not a
make fantastic
geologist.
signs over hundreds of millions of years.
But-
and this is just not supported by
I'm
the
not
small
a
incremental
geologist, but I
changes
have
that you're-
friends who are
that you-
geologists, and I have a question for you. Is uniformitarianism the governing, dominant, whatever word you want to use, paradigm of geology today?
I believe it is.
No, it is not.
Well,
Nope. And that hasn't been the case for like 70 years, 60 years. The dominant paradigm in geology today is called actualism. It's not uniformitarianism. It acknowledges that there are different rates of change and different processes that occur both temporally and geologically over time, and accounts for those when you're looking at how the past operated. Okay, so like uniformitarianism was kind of an analogy co-opted from geology into biology. It was never like a biological idea in and of itself in the first place. But even so, that hasn't been the dominant paradigm of geology since like the 1960s. Okay.
All right. All right. We still have the ability to look at biology today. And when we look at biology today, we see that animals, organisms aren't evolving. They have trouble when their environment changes. That puts pressure on them. They become extinct. We're going to see that all across the earth.
Extinction
this
happens,
year. Yeah. Extinction.
What happens historically after mass extinctions? This will be the last thing. Then we'll go, because we can just go forever. What happens historically after mass extinctions?
Well, after mass extinctions,
Yeah.
I'm not around to watch and see what happens after mass extinction. But
Well, as one of my friends is fond, we weren't there. Hang on. We were not there, but as one of my friends is fond of saying, the rocks were there. So what happens historically after mass extinctions?
We have animals.
Let
I
me.
believe that the answer I think is that there could be periods of recreation that God has placed different animals on the planet as at different times.
Okay, so the answer to the question, for everyone playing along at home, the answer to the question is a phenomenon called adaptive radiation, which is where you have lots of available ecological niches, and you have rapid diversification to utilize the available resources, right? So we see this most famously, for example, when approximately 65 million years ago, the non-avian dinosaurs went extinct, and then we had an explosion of biodiversity of mammals, filling those ecological niches like apex predators that were previously occupied by dinosaurs. We see this over and over. We also see this, for example, when flowering plants appeared. We see tons of animal diversification, mostly in insects. We saw this, interestingly, with whales. Once they got in the oceans, boom, explosion of diversity of whales. Yeah, you laugh, but it's right there in the fossil record. And you agree with the age of the fossil record. So, like, there you go. If it's your position that God did it, that God put those animals there, caused that diversification to happen, you need a mechanism, you need observations, you need predictions. You don't have them.
No, I don't. You're right, Daniel. I don't have a mechanism, but I do know.
I do.
That I do know. Well, I do know that design is design. That the information that's built into genomes
doesn't change, right?
The information.
Except that never is observed in nature, because Hardy-Weinberg doesn't happen.
The information in genomes is not generated by atoms and molecules randomly forming to create that information.
We can document cases where like it literally is, but that's okay. All right, let's go to the questions. I got a bunch of questions here. I know we've been going for a while, so let me ask you, are you cool to stick around for another like 10 or 15 minutes?
Sure.
Cool. All right, appreciate it. I know we've been going for almost an hour 45 here. So, folks, let's go to the chat here. This was Kyle Epp. Kyle asked, Jason, are animals alive today that weren't alive in the past? How did they arise? Were they not born, just created? Did changes accumulate over time? Do whatever you want with that. I think we kind of answered that a little bit, but go for it.
Sure. I think animals that are alive that were in the past that are not alive today. I think that I believe all of life in each genome, the amount of information is just incomprehensible. And those genomes are not created by random processes of abiogenics or atoms and molecules that are non-living forming together to create information in genomes. So
Origins of us, folks. There you go, on your bingo cards.
Information requires intelligence. So there.
No, it doesn't. That's wrong. I gave you an example where that's wrong.
Oh, well, you're wrong. Information absolutely does require intelligence. Information is not created apart from intelligence, Daniel. That's
Promoters,
the only place we're
randomized
getting.
promoters, functions promoters.
The
Like you could
the
randomize 100 nucleotides and you'll get promoters from your pool of random sequences. That's information from not intelligence.
the give me information in the genome that that can be created from molecules in an abiotic environment.
You don't have genomes in abiotic environments. That question doesn't make any sense.
Okay, well, you have genomes, and genomes are created with information, and information does not come out of an
That assumes
abiotic
genomes
environment.
were created. How are you quantifying information in this context?
Instructions instructions that lead to a specific action by a chemical process.
So that sounds like activity or function, but how would I quantify that as information?
It's you would watch and see what you would watch you would watch the outcome, you would watch the outcome, the output of the mechanism.
So 100 nucleotides, so 100 random nucleotides that can't function as a promoter versus 100 random nucleotides that can function as a promoter. The promoter has more information than the non-promoter.
What I'm saying is that the nucleotides themselves are created with information. And
Okay, but like if I have two sequences of 100 nucleotides and I tell you one of these is a promoter and one of them is not, how can you quantify the difference in information between the two?
Daniel, I don't know the answer to that question, but I do know that all genomes are created with information, and information is de facto proof of a design, an intelligent design, because information is not created.
You are welcome to believe that. All right. This question from Scientific Origins. I think we covered this. I don't know, Austin. I don't know if you were here at the beginning. But Jason, do you believe in a literal Noah's flood? I'm assuming the answer is no, but tell me if I'm wrong.
Well, I think if you am I not correct that there is a flood narrative in cultures all over the world? I think there's like 300 different
not all
cultures have 300 different cultures have a flood narrative. So I believe there was an ancient flood.
Oh, okay. That's interesting. I would love to hear. It's late, so I'm not going to ask you how you reconcile that with like extant biodiversity, but that would be a whole other 2 hours. Because the population genetics of that get really fun, but I would not get an answer. Again, from Scientific Origins, Jason, are you aware ID/creation propaganda mills admit to absolutely prioritizing dogma over evidence in their statements of faith? Which do you go with? Yes, this is referring to like Answers in Genesis, having everyone that works there has to sign their statement of faith, and you can't publish for them if you don't adhere to their statement of faith.
I am not. What was the organization again?
That's Answers in Genesis, which is a young Earth organization. So you don't necessarily, you don't have to buy into that because you're not a young Earther.
I'm not surprised that there would be a statement of faith required to work for Answers in Genesis. No, I'm not surprised.
No, I'm not surprised either. All right, Austin is all over here. Whales are artiodactyls. Yeah, you don't need the kind of changes you're talking about for evolution. Marine mammals alone destroy evolution. Yeah, I kind of agree with that last statement. Yeah, basically this is what we were saying before, where to go from like a terrestrial tetrapod artiodactyl, like some kind of deer or something, to a whale is actually not that much evolutionary change because you don't need new stuff. All the parts are there. You're just changing that's basically what this is saying.
Yeah, I know. And it's not
and I know that wasn't a question. Okay.
Not true. Takes huge changes to go from a land animal to a whale. And it takes more changes to go from a shrew to an elephant or gorilla.
They all got the same parts too.
Daniel, again, when we look at nature and we see the progeny of shrews, they're always going to be shrews. I know we've covered a lot tonight.
Algae or not, multicellular algae, we can go I mean, there's countless examples where, like, by any reasonable standard, it's not the same thing.
Yeah,
due to cladistics, right? And monophyly, it's always going to be within that clade,
the single-cell
right?
organisms
always going to be fish, vertebrate, all the way down.
And the single-cell organisms are always single-cell organisms. You do what you want in the lab.
I mean, that's literally not true, though, because we can watch them evolve in the lab. Well, here we go. Here's perfect to actually, hang on, sorry. I skipped this question. Kyle, I'm going to come back to you because this is perfect for Isabella Traster asked, Jason, What do you think is so special about a lab that allows a process to happen that can't occur in nature? So this was popped up when we were talking about the algae, for example,
Yeah,
right?
that's a great question.
Yeah,
So
like
so you have an environment where you are intentionally restricting all of the characteristics of the environment that the organism naturally lives in, and you're excluding those from the lab, and then you're introducing new characteristics that the organism never experiences.
they never
And
experienced predation.
No, the
Algae never experienced predation in nature?
well, that wasn't the example. I was thinking if she was talking about lab examples. I was thinking about
Yeah, the algae was a lab example. We
I was
evolved
thinking about
multicellularity in the lab. Yeah.
Yeah.
By
Right, okay.
exposing them to their natural predators.
And then in nature, do we see single cell algae evolving to become multicellular algae in nature in the ponds, or do we see yeast in its natural environment, other than in a lab, ever becoming multicellular yeast?
No,
In
well,
fact-
the
in fact,
yeast was
never did become-
sedimentation, so they don't typically have centrifuges in nature.
Thank you. That's it. And so you have to create. You have
But
to create
what's
a
to
special
stop
environment.
genetically? What's to stop these changes from happening? Because again, it's just all genetic and phenotypic changes. So the question is, what's to stop? Like the difference, I can tell you with the algae example, the difference specifically is that the density of the predator is much higher than in nature. So it's a much stronger selective pressure, right? In nature, the carrying capacity of the predator is much lower. So you never get to the strength of selection that's going to cause obligate multicellularity to be more adaptive than facultative colonialism. In the lab, you can impose a stronger selective pressure. What's to prevent that from happening in nature, hypothetically?
The reason it does, what prevents it happening in nature is that those organisms are really, really happy being single cell organisms, and they're finding a way to navigate
Personifying
life.
biology.
Well, it happens. So those organisms are finding a way to adapt to their environment as single cell organisms. So there's no incentive for them to become multicellular.
What if the host
And-
density
and-
was 10 times higher in nature? What if that was a thing that happened? Is there a barrier that would prevent them from becoming the obligate multicellular structures we saw in the lab?
The barrier is that they're single cell organisms, and single cell organisms don't become multicellular.
We literally,
That's
but
the
here's,
barrier.
okay, but you're missing my question here, right? The difference in the lab compared to nature was that the selection for being big was much stronger because there was a higher density of the predator, right? You follow what I'm saying?
Yeah.
Right? Hypothetically, in nature, if the density of the predator was higher, what mechanism prevents the same multicellular phenotype from evolving in nature?
It's hypothetical. That's what prevents it, because it's not seen in nature.
So there isn't one.
There's your
Like-
it's your hypothesis.
Because they
It's a
didn't
hypothesis.
so they didn't do any genetic engineering to those algae, right? They didn't like
And they didn't become multicellular. They're not.
they
They're still-
literally did, sir.
No,
They
they're
literally did.
They're single cell algae.
What's
that-
the
that-
word for a structure that has 8 cells and it can only have it must have at least 8 cells to survive, and only some of those cells reproduce? What's the word for such an organism?
Algae.
Is it unicellular?
It's a unicellular creature. There's
What does the prefix u? I feel like there's a disconnect linguistically here. What does the
No.
prefix uni mean?
It means single cell algae. Will
It
always-
means one, right? And how
That's
many
right.
cells are in an 8-celled structure?
8 cells clumped together of an individual.
They're not clumped. They're not clumping.
yeah,
We talked about it. They're clonal. It started as 1, it divides, it gets 8. It's not just teaming up. Right? It's not.
Yeah,
I think creationists have lied to you about that paper. I think you have been lied to about that one.
No, I'll go back and look at that paper. But the genetic material of each algae, do those 8 cells share one genome?
They are clonal, yeah. So they come from a single cell.
They come from a
right?
single cell.
And then they divide into 8 cells. So they're clonal, right? Just like we are. We're trillions of cells. We're clonal. Same idea. Yeah.
Okay, so there are not 8 nucleuses then.
Yes, there are. Yeah, it's 8
Okay, there are
cells.
8
yeah,
if
each
8
individual
nucleus-
cell has a nucleus.
Then they're each
yeah.
individual cells. They're not a multicellular organism. If each
Are you?
individual cell,
Well, you're 30 trillion individual cells. Are you a multicellular organism?
I am, but they're not a human. They're an algae. The algae,
Right.
those algae each have that single cell. They do.
And
Let me ask Daniel. Daniel, let me ask you a question about that. Do those 8 cells have different DNA that, or are they just clones of that single cell? Are they clones of, or do they have different DNA? Because my 37 trillion cells each have species. They each have special functions. They're
right.
differentiated. They're differentiated.
Wait, hang on,
They-
hang on,
they-
hang on, hang on,
they-
hang on.
they-
I gotta,
they
I gotta
access-
ask, I gotta ask, I gotta ask. Is it your understanding of multicellularity for humans that your cells have different structures and morphologies and functions because they are genetically distinct from each other in some way?
The cells of the tongue are not the same as the cells in the fingertips or on the cuticles. So each cell is differentiated in its function. So
No, no,
the
no, no, no.
cells,
I didn't ask if they're differentiated in their function. I asked, is the differentiation in their function due to being genetically distinct? Like your skin cells, are they actually genetically, do they have different genes or different alleles compared to your muscle cells or your neurons that allow them to do that thing?
they access a different part of the genome
That was not, no, no, no. Well, like,
No,
just
so I'm
answer
not-
the
I'm
question
not-
clearly. Like, are they clonal? Are they clones of each other that express different genes, or are they genetically unique?
they aren't clones of each other. Each cell has its own access to the nucleus of the cell that causes it to reproduce. So I guess the
I
answer
feel
is,
like
Dan-
you're not, I don't know if you're not
no, no,
understanding
no,
the
no,
question
no,
or if
no,
you're
because-
refusing to answer. I
no,
can't
it's not.
tell.
It's not. Each cell when it reproduces.
If I
through
sequenced a
mitosis-
skin, if I was to take a skin cell from you and sequence its genome, and then I take a white blood cell and I sequence its genome, are those two genomes going to be almost entirely identical aside from mutations that occurred since the zygote? Or are they going to be distinct in some way that accounts for their different morphologies and functions?
Well,
Is it the first thing or the second thing?
They're I don't know.
Okay.
But that's okay. That doesn't.
No, that
doesn't
is
change.
okay. It's just like I would appreciate. It's just in a multicellular organism, all of your cells are clones from the zygote. You have your zygote; it's one cell, and it divides, right? And there are mutations that occur. But if you take any two cells in your body, almost all of those 6 billion nucleotides in each one are going to be identical, right? They're going to be the same,
right.
And the differences across those different cell types are due to differential gene expression in all those cells.
Right.
And I just, once again, find it amusing and entertaining and just, you're not the only person to do this, but this is like a basic thing in biology, and you're sitting here talking to an evolutionary biologist telling me that I am wrong about the entirety of my field, and there's like very basic things about biology that you just don't know, and that's fine, but like maybe learn the basics before thinking you can like overturn the entire field. I don't think I'm out of step making that suggestion.
Yeah, I don't think I'm out of step in looking at nature and the way that mutations operate in the genome and the genome's preference to have mutations repaired because
have you heard me say at any of the points I've said it so far that there is not a generalized preference for that because there are mechanisms that operate in the other direction?
Oh, there's a generalized preference for it because the mechanisms that operate in the opposite direction, Daniel, are a very small slice of organism life. That's not the rule.
Considering,
It's the exception,
I think it's fair for me to ask how you're making that consideration, considering I explained several of them to you that you hadn't heard of 2 hours ago.
Yeah.
So how are you making the characterization that they represent a minority of what's going on when you didn't know they existed?
Because
you could just say fair enough.
No, it's not fair enough because
Okay.
because
It's your day. It's up to you.
because the the bacteria that you've talked about, the E. coli is always going to be an E. coli. Will always be
Yo,
an E.
now we're
coli.
just going in circles. I'm going to the next
Well,
question. All right,
you
you're
know,
just doing the Kenhoven thing. We're going in circles. Jason, have canines always been around? If not, how did they first show up? Take it away. How long ago? Yeah. Have canines always been around?
I don't know, but I think that it's a very interesting question, though. I've always been around. Uh, the idea that dogs
I don't
have
know is a
evolved from
perfectly
wolves.
fine answer.
Well, yeah, I don't know how long dogs have been around.
Okay.
I do know that the genome has hundreds of breeds, and there's a limit on the change that we see in the genome. We have everything from teacup poodles to Saint Bernards, and
That's
the
fine.
genome's not able to express itself in more differentiation than that, apparently.
I mean, that's not true, but that's fine. How can stasis be beneficial? This is a really good question from Petite Flower. How can stasis be beneficial when environments are constantly changing, whether that's climate change or competition or whatever? Yeah, ambient conditions are always changing, so how can stasis be beneficial? Yes.
Right. Well, I think stasis is. I can answer that too by going back to the E. coli. So,
But don't go back to E.
talked about-
coli. Like the E. coli we talked about, that's stabilizing selection in a very consistent environment. When we're talking about environments that are changing or environments that are dynamic, how is stasis beneficial? That's the question.
Yeah. And the answer is the same answer we find when we look at the fossil record and we see animals have genomes that don't change and they become extinct. And that's exactly what we see today.
So the answer is it's not.
No, it is for the environment that they're in at the moment. And if that environment changes, they have a very difficult time in adapting to that new environment, and they do become extinct.
I'm going to ask a follow-up question.
That's why we no longer have dire wolves.
I'm going to ask a follow-up question. Don't mechanisms exist that promote new variation in the face of changing environments?
There may be some mechanisms to do that. It's a.
You can just say yes.
Well, yeah, okay.
Because the answer is yes.
Okay, the answer can be yes, but the answer is usually no. Okay.
Well, the answer is yeah. Like the mechanisms exist. It wasn't like a degree question. It was a do such mechanisms exist, and the answer is they do.
Yeah, and they're not efficacious in the total picture because animals are going extinct, and we're going to see many more animals become extinct than we're going to see truly new species come on the earth. So the answer is that stasis is the rule.
And now we're back to like species fixity. This is something else. All right. Who designed the human shoulder? It's just awful engineering. The human eye is perfect. As trichromatic creatures, we are inherently colorblind relative to the lowly mantis shrimp. Yeah, okay. Talk about all these perfect designs. They're not.
That's they are.
Point. Ankles are my go-to. I hate ankles.
Okay, can you got just doggone it. I wish I have a. Can I disappear for 1 second?
Be my guest.
Can you give me just a minute?
Be my guest. Chat, these are very good questions, by the way. I appreciate all of you. These are excellent questions. Like these are not trolly questions. These are legit questions. I'm gonna save that one right there. Cheetah, that's a good point right there. I'm gonna save that one. Yeah. While Jason's going to get something, I'm just going to go through some of these other comments. Templar Warden, I hope you don't mind me conveying this. Someone lied to him about those papers. Yeah, I agree. Let's see. This made my night. Sorry, I was just going through some of the other comments while we were sitting here that I had starred. Dr. Daniel Sternial Cardinelli. That's really funny, Isabella. I think that's really funny. Okay, we were up to this question right here about the human shoulder, and we're almost at the end, I think. Yeah. What do you want to show us about? I mean, you could do all the like human engineering is really bad, but Nathan Lentz is actually good. Nathan Lentz wrote a book on this that I recommend. There's like, you can redesign the human body in so many different ways that make it not prone to all the problems that it has. So yeah. Spine is another good example. It's like you take a suspension bridge and stand it up on its side. Sorry, go. What is it that you grabbed?
I couldn't find. There's a book. There's a book recently written by an engineer in the United Kingdom who has he's helped design the international parts of the International Space Station, and he has designed the cycles for the UK's cycling team, and he's written a book about.
And what's his training in biology?
He's an engineer.
Exactly.
Yeah. Okay. And he knows great designs when he sees it. So his example, Daniel, before we leave this question, he says that the designs of the human joints are better than any design of joints that he's seen in any manufacturing environment.
Okay. That's fine. That's fine.
My point is the human body is wonderfully designed. It's not poorly designed.
I don't think an engineer pointing out that joints are well designed, I don't think it follows from that the human body is designed. You know what I mean? Like, I don't think A, therefore B, I don't think that one flows.
Well, it does flow.
So let's go to, does bacteria becoming an organelle inside a eukaryotic cell count as bacteria becoming something other than bacteria? That's a good question. Yeah, does endosymbiosis count as a bacterium becoming something other than bacteria? Yeah, that's a good question. Yeah.
Yeah. Well, I think it shows that it adapts very well inside of a eukaryotic cell.
But it loses most of its DNA, so it's actually not beneficial for the bacterium.
Well, we see. I guess that it goes back to the things we see in nature that don't really make sense from an evolutionary point of view, and for the individual, like the like the spider that is eaten by its mate. So there's the spider dies, but it's supposed to be.
I can explain that for you. That's super easy, PopGen. That's super easy to explain. I can totally explain that example. So that's called that is called nuptial cannibalism. Right, yes. Where they mate and then the female eats the male. And right. Do you know the underlying PopGen for that? Because it's super easy to explain. It's just game theory.
Okay, let me run with this for just a minute.
So yeah, here we go. I'll explain it to you. Here it is. Here's the thing, because no, wait a minute, hang on. I'm going to do it. There's a word you need to know. There's a word you need to know. Yes. The word is semiparis. Do you know what the word semiparis means?
I don't know what it means.
That explains the whole deal. Semiparis means you only reproduce one time in your life. So if you're semiparis and you're a male, right? You're so girl spider, boy spider. Boy spider is going to die after it. Like he's not mating again, right? So now, if you stick around, who are you competing with? I bet you can get there. If the male spider sticks around, who is the male spider competing with for resources after he mates?
Yeah, he's competing with the spider he's going to mate. He can't
competing with the mate, and ultimately who else? Competing with the kids, right? And now if I am competing directly for resources with my kids, does that make my kids more likely to survive or less likely to survive?
We'll make them less likely to survive.
Less likely to survive, right? Which means we're going to see selection to minimize the competition, right? Right? Selection is going to minimize that competition because that makes your kids more likely to survive and therefore improves the representation of your genes in the next generation, right? That makes sense? Cool. So let's follow that logic. Let's follow that logic. What else could we do to make it more likely that our kids survive, right? We're going to
reproduce more often.
Hang on. That's not an option though. So look, I don't want to mute you, but I will because we're going to do this. Just let me explain it. Because reproducing more often is not on the table, right? That's not an option. You are what are called an R-selected species. So basically your point is to mature to adulthood as fast as you can and have as many kids as you can as fast as possible. The trade-off with that is that you only get to do it once, right? So mating multiple times, not an option. So we know that we don't want to compete with our kids. We can do the PopGen on this. We can just directly this is easy. Now, what's another way we can ensure that those kids are more likely to survive? What if we provided more resources for the female? Because more resources means bigger eggs prior to fertilization, means more likely to survive. And this is a thing that many arthropods provide. It's a thing called spermatophores. It's sperm with food. so that the female eats and then fertilizes the eggs. The eggs are bigger, they're more likely to survive. That makes sense?
Yeah.
Yeah? What's
Yeah.
another source of food that the male has at his disposal? Himself. So what happens when they mate? It's actually not the case that the female kills the male. What actually happens, if I can kind of demonstrate with my hands a little bit,
Sure.
they kind of mate, and then the male, after he mates, like flops over and impales himself on the female fangs. That is the most adaptive behavior because it maximizes the likelihood that their kids survive. It provides additional energy to those developing eggs so that they're bigger and therefore more likely to survive. And it minimizes competition once they hatch, right? So that trait is super easy to explain in the context of population genetics. That one is so easy, and I love teaching it every year. Yeah, there you go. All right, moving on. We've got Mr. EPF. You keep asserting that we don't see evolution happening inversely. Where can we see a God creating and interfering to prevent evolution? Yeah, that's a good question. We don't see design or creation.
You know, thanks. I'm getting tons of questions, and so let me ask one to the group, if you would.
Please.
You know, evolution is a fact. Let's assume it is. Assume it is, and if I understand evolution correctly, right? And tell me if I'm wrong, but evolution doesn't create anything out of whole cloth. Evolution is always modifying something that evolution is evolution not generally modifying something that previously existed to get.
I would say generally, but there's a big difference between generally and always. That's
okay. All right.
big difference.
Generally, evolution just modifies something that's previously existing to get something that's more adaptive currently. We have literally a million different organisms on the earth. And if evolution is a process that occurs, it's occurring all the time. Do we see organs that are being slowly modified
Yes.
to become different types of organs?
Yes, we are. Yep.
I've asked that question. I've asked that question of AI
We've literally
programs.
witnessed things growing new bones that didn't exist before.
Okay.
That's a new thing. That's a new thing,
I think
right?
I think that's something you generally don't see. You don't
Generally,
see,
sure, but we do see it.
Okay. It's if evolution is a process that's currently operating in nature, I believe we would be able to see evolution happening in species we observe today, and we would see the changes midway among organisms, and you just don't see that. If it's a very slow process, there should be thousands of organs, thousands of species that are undergoing some sort of evolutionary change. We just don't see it. The genomes,
I mean, we do, though. That's the thing. The fact that, well, we talked about Hardy-Weinberg equilibrium before and how it's not like a thing we observe in nature, right? We talked about how this doesn't happen, which means that everything is evolving. If it's not at equilibrium, it's evolving. It's one of the two things is happening. Now, you could say, well, that doesn't count because we're not seeing new organs evolve, right?
Yeah.
Okay. On what time scale do you expect to see a new organ? Do you think like something without a placenta is going to give birth to something with a placenta, right?
I would point to nature and say, if I have a million organisms and evolution is actually happening throughout nature today, Daniel, the whole idea.
On what time scale do you expect to see this happen, though? That's a fair question.
I would think that at any point in time with a million species, you're going to see species that are being intermediate to some stage of evolution that has there's no way to define what evolution would look like when there's no.
You don't think like a lungfish constitutes some kind of intermediate between marine and terrestrial?
It's like a platypus. We have very unusual organisms that are they're just different from like we don't. Daniel, as you know, there's no pre-bat in the fossil record. We just have bats. It's just there. And if there was a pre-bat
Yeah,
We just see
do they
it. There's,
typically live in areas? Are bats typically likely to like subject to sedimentation? Like the fossil record is very rich in
yeah.
aquatic organisms, marine organisms, and organisms that exist in like intertidal areas or marshy areas, right? But it's harder, not impossible, but it's harder to find things that
Well, we have fossilized bats,
we do have fossilized bats. We
and
don't have
they're
much
nearly
of
identical to modern bats.
that. Well, that's not true. They're not identical to modern
I didn't
bats in
say
the
they
same
were.
way
I
that the
didn't say that.
fossilized coelacanths are not identical to modern coelacanths, or the old crocodiles are identical. Right? They're not. They're the same basic ecological niche, but they're not identical. Like, let's be
More
clear.
similar than a teacup poodle is to Saint Bernard, which are the same species.
No, I'm not giving you that one. No, because those both have legs. You know, it's no, I'm not giving you that one.
They both have
All right,
wings.
we're going to be here all night if I don't get through these, so let's get through them. I told everyone I would do it. Austin wants to know if you would like to be in an open panel of, if you want to do this again with Austin and his friend Mike, who are both very nice people. If you want to chat with them, I can, if the answer is yes, I can hook you up via email. So let me know. All right, I'm going to move to Kyle. I've never observed God interact with genes. Fair point.
Okay.
Could you define information? I think we
In-
kind of asked that.
Coded instructions that give purpose to an organism.
And can you quantify it? Like how much information is in your genome compared to a chimpanzee genome?
I can't quantify that. I think
Fair enough.
that may be
That's fair.
You know, that's very interesting. I think the
This is-
the genome for wheat has much more information than the genome for humans.
It's bigger
That's a-
genome.
Yes,
Have
yeah.
you considered turbo adaptation? That's not a serious question. That's a joke from like a recent thing. Don't worry about that. That's a joke. Thank you, Sydney, but that is a joke. Okay. I used to believe in intelligent design. Now I'm an evolutionary biologist. How do you think that happened? Conspiracy theory equals bad arguments. That's
Okay.
fair. Yeah.
Okay.
So, in other words, do you think Austin was convinced by the evidence, or was something else going on?
I think he was convinced by the evidence. Austin, I would like to ask: does it bother you as an evolutionary biologist that we have reptiles and we have amphibians and reptiles, but there's no group of animals that's intermediate between reptiles and amphibians, and you know we have tens of thousands of species of one. We have 12,000 species of the others today.
I will
But
say,
there's
Austin, if
no-
you want to
there's
answer
nothing.
that, I'll read your chat. It doesn't have to be a super chat. Just let me know you're answering, and I'll read it.
Okay.
Yeah, if you're still here, Austin.
Not a problem, Austin.
Here you go. I'll let him answer that. If seeing hippos only giving birth to hippos today is okay to extrapolate, why are changes we see today not okay to extrapolate? That's a fair question.
Well, I think the changes you see are changes on the periphery. We don't see any changes today that are significant, that are going to change humans to ever be anything but humans, shrews ever anything but shrews. hippos
Unicellular
ever
algae to
anything
multicellular algae, animals, a defining characteristic of animals being heterotrophic. We never see them becoming photoautotrophs. We actually do.
Yeah.
You know, reptiles laying
Yeah.
eggs, they don't give birth, except some of them do, right?
Yeah, and every one of your examples is an example that's going to allow that organism to remain on the earth, operating and being productive.
In other
as
words,
that
natural selection works, yes.
as that organism. So
Yes.
that particular type, that reptile, will never be anything but a reptile. It's not going to evolve to become an amphibian, and it's the genomes are fairly fixed, and mutation repair mechanisms exist because they recognize mutations as not being helpful to the genome.
Austin, I don't see, if you could tag me if you want to answer that question so I can see it, that would be good. Thank you. You can keep saying we don't see this stuff, but that's fine. Do you think you have, this was from when we were talking about multicellularity before. Yeah, I was confused by that too, but I think we worked it out. Thank
No,
you,
every
Kyle.
cell, I would say every cell has its own nucleus. Is what I would think.
Can you explain how the recurrent laryngeal nerve in giraffes is good design?
Yeah, I've seen.
know that one?
I know that one, and I know.
Yeah.
I think the answer to that would be, you know, we the design in nature is quite incredible. There's a reason that humans don't design with DNA. We can't. Nature
Okay, but the
designs with
question
DNA.
was, is the recurrent laryngeal nerve good design?
My point is, we don't have the perspective of
I mean, you
designer
could just say
to know.
no.
Well,
You can just
you can't.
acknowledge that it's dumb
No,
because
I
it's
can't.
12 feet to go 12
Yeah,
inches, right? You can just
right.
admit that. There's nothing wrong with that.
Daniel, we don't have a creature. Then get into the embryo as it's developing, and reposition it. And
That's
just
the
see how
point,
well
though.
that comes
That's
out.
exactly sir, that is exactly the point, is that it is developmentally constrained by its evolutionary history, which is why it evolved that way. If it was designed separately from, for example, marine gnathostomes, which is to say sharks, right? If it
Daniel,
was designed separately, then you would have been able to unlink the nerve from the blood vessel. But it wasn't. It's constrained by its evolutionary history. Yes, the thing you just said is exactly the point. Yes.
And we weren't there to see it be designed, and so we can't say that we
Correct.
know we know when we're looking at the design that it's a bad design, because we don't
We
know
can just
when-
objectively do that because it costs extra energy and it takes extra time. Like, yeah.
then go into the embryo, change it, and see how well the giraffe does.
It would be
I
fine.
think the
The
giraffe-
point
I
is,
think-
it's
I think the
developmentally
giraffe- I think-
constrained, so it wouldn't happen because of the way the tissues
then you
develop
don't know that it's a
interlinked.
bad
Like, that's the
design.
point. Okay.
Then you're assuming
Okay.
it's a bad design
We
without
got
being
actually. Let me do that one last. I just got this is Austin's answer to your question. In a word, no, it doesn't bother me. I heard all these arguments when I was still an intelligent design believer and found them to be wrong or specious. That's the answer to the question.
Thanks,
Okay.
Austin.
This last one, this is kind of a joke. I think we're gonna call it here. This is kind of a joke. Dan, I think you were Jason's Huckleberry. That's a good movie. Jason, how do you think you've done in this conversation? I'm curious if you think you countered Dan's argument. That's a good way to end it up.
Okay, I think that's a fair question. And
Fair question.
every time Dan presented a method of an adaptation of an organism, he helped that organism become a better organism as that organism. He never presented a structural change to that animal that's going to make that animal ever be anything other than the organism it is. So if it's even a bacteria that is benefited by mutations that it generates itself, it is adapting to its environment as that bacteria. So we don't see bacterias with if there's speciation. Much of that speciation is not helpful to the organism. Sometimes it reduces its ability to reproduce with its descendant or with its ancestor, with the ancestral populations. So that doesn't necessarily make it a better organism. And if organisms do benefit from bacterial mutations, they are actually existing as that organism, as that bacteria. So that E. coli is never going to be anything other than an E. coli. And the other evidences that he provided, he has many examples that are on the periphery, small changes like the digestive, humans being able to digest milk. That's a beneficial mutation. What Dan never did address, though, was the more common effect of mutations that are beneficial is typically they're not just beneficial. More often than not, there's antagonistic pleiotropy, where there's some offset to the benefit, so that the organism actually isn't improved. And if it carries that mutation, it's carrying something else it wish it hadn't carried. So there's a mutation that gives people a resistance to malaria. It also causes sickle cell anemia.
There's also a variant of that doesn't cause sickle cell.
Okay. There's also a mutation that causes bones to be more robust, bones that don't break as easy. It also results in facial deformities and pain, muscle pain, and muscular pain, tendon pains. So that's typical of mutations that are identified as being positive, as being good mutations. They come with some offset, and mutations never affect just one gene. They always affect more than more than one gene. So they're not a mechanism by which, and there's, as Daniel's got his smug face on, and I understand,
I have no poker face. I have
yeah,
no
yeah.
poker face in these things. The expression that I learned a number of years ago from somebody else, they said of themselves, "My face has no indoor voice," and my face has no indoor voice. So you're going to know what I'm thinking of your answer as you're saying it before I say a word.
Yeah, okay.
Because
And, at the end of the day, Daniel, there's a reason that you and I both love our children, and we're really thankful that we found our children born without mutations. If somebody were to if somebody was to walk in the delivery room, Daniel,
we all have
say,
mutations.
Daniel, yeah,
They do have mutations. So do yours.
yeah, the kind
It's
of
true.
mutations, that cause some type of morphological change.
Yeah. Yeah, my wife and I carry a severe genetic disorder. Yeah. We have to make sure our kids were heterozygous or homozygous dominant. Yeah.
Yeah. Okay. All right.
Everyone carries mutations. Yeah. Everyone carries one to a handful of recessive genetic conditions. Yeah. That's just a thing.
Right.
So-
And so those are variations against a genome that once didn't have those mutations, and we would rather not carry those mutations into the future. So we can talk about the benefit of mutations as we admit
want them
here
to
in
have
this,
some mutations.
as we admit that we would rather our children not be born with mutations. There's a reason that those repair mechanisms.
That's wrong though, because that's why sexual recombination exists. Sex has a lot of downsides, right? It's slower, you make fewer offspring, it spreads disease, it costs a lot of energy. You wouldn't do that if it was better to have clones. The point is that having kids that are not clones, which is to say they have genetic differences, is beneficial.
Absolutely, mutations aren't.
Well,
That's
you
why
say absolutely, but then that disproves the thing you just spent 5 minutes saying.
No, Daniel, I started the conversation by saying homologous recombination is a good thing. Okay,
Right, which disproves the thing you're arguing. I literally had that on my list of things to say. And then you said it for me. That's because their whole point is stasis, but then recombination exists and is harmful if you don't do it.
Daniel, stasis in terms of the sequencing of nucleotides not changing. That's
what do you think recombination does?
It doesn't change the sequence of the.
It literally does. Your kids have a different sequence of DNA compared to you due to recombination.
Those are not mutations.
Right. It's a different mechanism of generating new variation. Yes.
And mutation is defined by a change in the sequence of the nucleotides. That's the definition. Daniel, if you don't have a change in the sequencing of nucleotides, you do not have a mutation. And
you have there are other mechanisms of change in your genome. It doesn't have to be mutation. Yes, there are multiple mechanisms. Yes,
and that's
true.
You're going back to redefining evolution as evolution when the species remains the same. So it's the same. We're humans. We're going to be Homo sapiens, and we're going to get all these mutations, and we're evolving. We're not evolving.
So I'm gonna real quick. I'm gonna point out two things. First, small thing, right there at the end, you're redefining evolution. No, I'm just defining evolution as it is defined. And if you think that definition is wrong, you're not arguing with me at that point. Now you're arguing with the glossary, okay? You have to take it up with that. It's not me. The second thing, and the broader point here, and this is where we're going to end it because now we've been going a while and it's late. You said we only see these small change, lactase persistence, whatever, all that small stuff.
There's stuff on the periphery.
You also did not, in your explanation there, mention what I think is fair to characterize, and you can disagree, but I think it's fair to characterize as larger scale changes, things like the evolution of multicellularity or photosynthesis in animals or changing your reproductive mode from egg laying to live birth. These are all things we've directly observed, and it can be your characterization that scale of evolutionary change is still within that periphery that you're describing. I think reasonable people would look at that and say those are quite significant evolutionary changes in terms of the evolutionary history of life on Earth. And if your position is that no, those are just changes on the periphery, we can document those, that's fine, then I'm perfectly happy, and I'll end this on agreeing with you. I am perfectly happy to accept that, because that means that you don't need large scale changes to get all the biodiversity that we see. As if multicellularity, endosymbiosis, those are like some of the biggest, most significant evolutionary changes that have ever happened. So if those things are just changes on the periphery, it doesn't fundamentally change what the organism is, trivial really, then that's fine. That makes evolution easy. Evolution was really easy then, and there's no reason to think whatever process, whatever change can't happen. So that's where we can leave that one. Folks, this is where we are going to end it. I want to thank all of you in the audience for watching this live and being very active, and also like not being rude. Like those were good non-trolling questions. Those were like legit questions that contributed to the discussion, and I appreciate all of you for that. I also will thank Jason for coming on when I said email me, and you can just come on and we could talk about it instead of going back and forth in comments. And I appreciate, Jason, that you did that. And let me know via email if you want me to hook you up with Austin and his friend Mike, and I'll connect you guys to set up a chat with them if you want. And I think that's where we can leave this one. So folks, before you go, please remember to do the algorithm-friendly things. Hit the like button or the dislike button. I don't really care. Leave a comment, subscribe, and I will just tell you that in terms of what I have coming up, I released for members a little teaser video today. It'll go public on Friday. That I have to correct something from my video from last week. I said something was horizontal gene transfer. It's probably not. It's probably a different thing, nuclear gene transfer. And I've gone down that rabbit hole, and I'm going to do a deep dive into that. So that's going to happen. I hope to have that done for next week. I don't know if I will, but it's going to be really cool. And then I have a couple other live shows in the works that I don't know if the guests are ultimately going to do what Jason did and come on, but I'm hopeful that they do. And as soon as I have something locked down for those, I will, of course, let everyone know. So that's where we're going to end it, everybody. Have a great night. And as always, don't get fooled. Thanks, everybody.
And happy no